so welcome to the first official part of the education program for this weekend. This is the research plenary. We have an amazing lineup of speakers. You'll see on the seat, every second seat should have a program. We have 14 speakers in this first segment. This is how we'll roll this morning. So it's going to be a little bit like speed dating for scientists. You will hear from each of the speakers in rapid-fire abstract research presentations. So in part one, each of the speakers will have 4 minutes to present their work. It really is the ultimate challenge for scientists who spend decades researching their work and are so deeply passionate about it. To try and condense it into 4 minutes, I think actually requires a Bill Bush motivational speech beforehand, because we got this, we can do this, come on team. We're going to roll it through. There'll be no introductions between speakers. They're just going to pop up in order as they appear on the program. The idea is not for you to get sort of the full in-depth. Obviously in 4 minutes it's very hard to get that from each of these speakers, but the idea is just to get a little snapshot of what they're doing, get an eyeball of who that person is, and throughout the course of the weekend, they're going to be here to approach, to speak to delve a little bit more deeply into the research that they're involved with. There'll also be a Q&A session at the end of part one, and then again at part two. So speakers, there'll be at 3 minutes, somebody standing up in front to let you know that that's time. And you will have to open up your own slide decks, but they're all here on the desktop. So just briefly before we start, there are many on-site research studies taking place over this weekend. Really encourage you to go and check that out. You can find out more on each of these either by visiting the conference website and going to the conference, so go to the BDSRA website and the conference page. You'll see there's research studies link on there, and you can click on each of these and find out more about it. Alternatively, go to the front desk and there'll be pamphlets around, and they'll let you know where to go for each of those studies. But as Dave said in the beginning, this really is this is your research conference. This is all about you and your children, and your participation really helps us drive the science forward. So without further ado, I will invite Dave up onto stage, and he'll kick us off. The trapdoor will open at 4 minutes, okay? So keep going.
So I'm changing hats here, no longer your MC. I'm going back to being the two things I do well. The first one is I'm an educated hooligan, so I will be standing up at 3 minutes from your talk, and at 4 minutes coming and tackling you. Coach Bush coached me a little bit on how to do that before we started. So, okay, so I'm Dave Pierce. So my background, just for those of you who are new, is I built a research program at the University of Rochester for Batten disease, wanted to do more, established a children's research center at Stanford to expand more on Batten disease, built a lot of the models that are being used right now. Realized this. There's a lot of rare diseases, and it's really hard to communicate the needs. So I use Batten disease as a platform for some bigger programs, and I oversee a large research consortium with respect to making sure we get more things done. I'm going first because you're going to have a lot of questions about the state of the affairs right now. You're going to hear some really good talks on clinical research and preclinical research, what clinical trials that are coming along. So you're going to say, what's the FDA doing? What's the NIH doing? So that's my kind of role right now. I have meetings with the NIH, FDA, and I was just with CMS. So the Centers for Medicaid are going to become more and more important in this world of rare diseases for us right now. Because if you are the smartest person in the room and you develop a treatment, who's going to pay for it? And that's a huge issue right now. So any opinion I say right now is Dave Pierce, and we all know I'm pretty friggin' opinionated. But just to give you the lay of the land, there's a new administration, okay? It doesn't matter, it's a new administration. A lot of what I'm going to say has not changed. It's just new people right now. And the new people at the NIH and the FDA and at CMS suddenly get inundated with 1,000 political parties because the patient advocacy groups who advocate for rare diseases, and it's really hard for them to see the forest for the trees. So part of what we need to do is, when I consult for companies, and I never take money, by the way, is I get asked, how many patients? Then we talk about the platform, the science, and then they will say, how many patients? So the move right now is to really emphasize that you can actually apply some of these clinical trials and therapies to more patients, because the bottom line is there's only three ways this gets paid for. Philanthropy, and you're going to hear a little bit about that. investment and people want a return on investment, they want to make money on this, or health systems need to adopt this, and their return on investment is cost savings, so you don't have so many visits to the hospital. Okay, so that's the strategy that they need to adopt. NIH right now, we're hearing that they're reducing certain programs. Okay, so that's just the lay of the land. That's the new world right now. You heard a lot about they made this massive cut on indirects. I will tell you, at Stanford Research, when I went there, every dollar we got 45 cents on the dollar indirects to pay for light bulbs and stuff like that. I got it up to 65. I kind of really pushed on that. Do you think it really was 65 when it was originally 45? Of course not. So when Harvard is bitching and moaning about losing their indirects, they just have to adapt because they want to have more money coming in. So we'll figure it out. There's going to be prioritizations. FDA right now. That's an interesting one because obviously I'm not going to get into the politics here. Who's going to tackle me? The politics here, but they're learning right now and they understand they're trying to make it easier. They were trying to make it easier before, but you've got 1,000 political parties pushing for this right now. So we're going to get there. They're very open to this and there's been some very good meetings. And then finally CMS. In my wacky world of Dave lives a celebrity life, I actually know Oz, for example. And he really wants to make a difference. So I've been having conversations with him. Dave, make it easier for us to understand the impact. Give us a payment model. Give us a payment model, which is really pretty wacky because I'm not even allowed a checkbook at home. But make it more understandable, and that's it. So I want you to take away just from what I'm saying right now. You may be hearing about what's happening with NIH, FDA, and CMS and rare diseases. The glass is half full, or the hands are up, or whatever. It's going to be a good time for us to advance the mission because these guys have got some really good science that's going to translate, and we are going to get it paid for.
Where are our files meant to be? Thanks. So I'm John Cooper from WashU, and my job at the beginning is just to give you a quick overview of what this session is like and cover some of the topics we're interested in. So we're all of us here because we want an effective therapy. We don't sadly have that magic pill, and the path to get to it is not a straight line. It's a tricky one. That's largely, it takes us too long, and it's largely because we're missing pieces of the jigsaw puzzle that we need to be able to solve that. In particular, we really don't know what many of the Batten proteins do, so it's tricky to figure out how they go wrong. So we use a bunch of different animal models. They might be cells, we'll hear about that. They might be animals, we'll hear about that. And we try and figure out what their disease is like and what we can then try to do to change that. So a lot of that work falls into the category. Dave used the word preclinical. These are experiments we do, trying out things that we hope will make a difference. We have two goals. One, to see if they are effective. Do they do their job? Second, are they safe? If that pans out, then we would go forwards and we would want to do a clinical trial. Now that's still an experiment. It's just an experiment done with people and has exactly the same goal. It's to see, does this work? And it's to see whether this is safe. And then we hope at the end of the day, we might end up with an approved therapy like happened for Brineura for CLN2 disease. So what are the obstacles we face along that tricky path? Well, as I said, we don't really know what many of those Batten proteins normally do. And although all the sorts of Batten disease are sort of similar to one another, they're actually not the closer that you look. So comparing them to one another, assuming they're all the same, isn't quite right. So there are many things we didn't know when we started this, like which parts of the brain or which cell types were affected. But that's important because it tells us where to put our therapies so that they can be most effective. And what sort of therapy might that be? We also have problems with our models, which is why we've had to generate some newer models, and we'll hear bits about that today as well. And as David's alluded to, there's a whole series of other considerations to do with funding, to do with the politics, to do with the NIH and the FDA that we have to deal with as practical problems to get to where we need to be. So what is it we want to find out? Well, they're very basic chicken and the egg questions. How, where, and when do things happen? We ask how our Batten cells or our Batten animals are different to an animal that doesn't have that disease. And that sort of measuring things that are different comes in lots of different sorts. It might be behavior, it might be molecular, it might be things we look at down a microscope. And we ask how these things change over time, where do they happen, when and how. And that, as I say, tells us where to put our therapy to try and be most effective. And again, we need to know, does that work? Does it make a difference? And is this safe? So what are those therapies? They fall into different sorts. One, we hear a lot about enzyme replacement, giving that back. We hear about gene therapy, supplying a working copy of the gene. We'll hear about drugs, small molecules that need to be developed. We hope to either stimulate something that's not working or block something that's causing a problem. We hear about stem cells. Stem cells used to be a treatment tool. Now they are a research tool that we use to ask particular questions. There's gene editing, and we'll hear a lot about the antisense oligonucleotide approaches. Now, all of these have technical problems. We have to make sure they work. And again, are they safe? So we only have a brief snapshot today. There'll be a lot more that's coming at the conference in Australia. That's coming up this fall. And I'm also waving a flag for a conference I'm organizing in a year's time, next August, which is the 200th anniversary of Otto Christian Stengel reporting what we now know as CLN3 disease. That's going to be in a small town in Norway where he found it. I have flyers. If you're from a company, you can sponsor us. We'll look forward to hearing from you and receiving your checks. Okay, so this is me being my postdoc ever from Poland. It's a tough thing to do. So I'm going to talk about what's happening with Batten disease outside the brain, and as a warning, that will involve poop. So we're very interested in what's happening in Batten disease. We think mostly of the brain as being really affected, so that's where we try to put our therapy, whether that's enzyme replacement or gene therapy. An example, we're doing gene therapy in CLN1 sheep, and that's going really well. We're getting some promising results. We've had some great results doing gene therapy in CLN2 mice, and that completely stops the seizures that normally kill those mice. However, even though we've done a great job of treating their brains and they're not having seizures, half of them still die. And that's because they have disease somewhere else in their body that's really important. It's life-limiting, and we need to try and treat it. So I'm going to tell you a bit about the things we're looking at in terms of the bowel and in terms of nerves and muscles. So this is all family-led research. The reason we started looking at the bowel was because you told us we really needed to do that. We know there are bowel problems in all the Batten kids. There's bowel problems in our mice. They are full of what I have learned the technical term is fecal material. They are backing up and filling up because things don't move along. What's important for things moving along is the second brain, something called the enteric nervous system. There's 500 million neurons in your bowel. So we know nerve cells in the brain die. Perhaps those nerve cells in the bowel would also die. So we tag teamed with some bowel experts to see, does this happen? What does it cause? And what could we do about it? So to cut a long story short, CLN1, 2, and 3 mice all have severe bowel disease. Things do go slowly. They back up. We know that's a problem that's intrinsic to the bowel. It's not secondary to the brain. It's really something going on in the bowel itself. What's going on is roughly half of those nerve cells in the bowel die, and something very interesting is happening with where those cells die and the bowel's immune system, a bit like what's happening within the brain. We also know this happens in children. It's not just some weird mouse thing, and we've been treating it. We've been treating it by giving gene therapy into the bloodstream, and we published earlier this year We can speed up that transport of stuff through the bowel. We can protect many of those neurons. And it does a good job. It doesn't treat their brains because that treatment doesn't reach it. So what we're doing now is treating both the brain and bowel at the same time. Now, the other area we're interested in is nerves. We all have nerves in the rest of our body. They help us feel things. Sensory nerves, they help us move. Motor nerves. There's a nerve ending on a muscle. We looked at those nerves, and surprisingly, the nerves are pretty much okay. The problem was at where their endings were. So we see fewer sensory endings. Our mice are more sensitive and then less sensitive to pain, and we're looking at that. We see real problems with where the nerves connect to muscles. There's a particular type of cell there that's really important. That dies off, and the nerve fibers die back. We think that might actually start in the muscle itself, and that might be causing all the problems. But the good news is, again, like with the bowel, we can treat that and improve it with gene therapy in our mice. The last thing to talk about is problems with swallowing, another patient-led study. This is a bit like a greatest hits of everything we've talked about already. Things to do with the brain, things to do with nerves, things to do with muscles, things to do with the bowel. We now have evidence, and this is all work funded by the Batten Disease Global Research Initiative, that this is definitely happening. We're making movies of how mice swallow, where the secret ingredient is Hershey's chocolate syrup, and we are treating this with gene therapy too. Now, I'm just the stand-up guy to talk about this. I have wonderful people in my lab, and you're about to hear from one of them in a moment. I say thank you to them. Thank you to all the very clever people we work with and the people who fund us. Thank you.
Hi everyone, my name is Katie and I recently joined, yep, that makes sense. Hi everyone, my name is Katie and I recently joined the Pediatric Storage Disorders Lab at Washington University in St. Louis. And today I'm going to be talking about the role of the brain's immune system in CLN1 disease. So you've probably heard a lot about the brain with Batten disease, and part of that's the death of nerve cells or neurons is sort of a hallmark of Batten disease. And accordingly, most research focuses on neurons and why they're dying. But as it turns out, neurons aren't the only cell in the brain. In fact, glia cells are the most common cell type in the brain. And so my research right now is focusing on microglia. So it's a subtype of glia, and it's the brain's primary immune cell. So they help respond to injury or illness in the brain. And we don't really know what microglia are doing in CLN1 disease. Through imaging or looking at the cells, we can tell that more microglia are turned on or activated in CLN1 diseased brain. And we can actually see that these microglia are being activated before neuron death, and they can help predict where neurons are going to die. Really interestingly, we see this increase in activated microglia in the spinal cord before the brain. So that really helps with learning about the progression of the disease. But we still don't have a great idea what they're doing. And so we can use sequencing to try and get a more complete picture of what's going on, what these cells are doing. So advances in technology allow us to better analyze what cells are doing, so we can look at healthy and CLN1 disease mouse models, and we can also look at treatment. We can see how these cells are responding to treatment by treating the disease mice with enzyme replacement therapy. And like I mentioned a minute ago, we're interested in the brain and the spinal cord, so we can start to see some of those differences, especially in how it relates to treatment. And we can sort for microglia. So there's a ton of cells in the brain and the spinal cord, and they're all doing a bunch of different things. So helping to sort and look at just one cell type, we can see what those cells are doing and how they respond. So we can sort for these microglia, and then we can sequence each cell, and we can try and get a good idea of what they're doing. And the first big thing we see is that not all microglia are the same. So we've already sorted from all the cells in the central nervous system to just microglia, and now we can see that even within microglia, cells are doing very different things, and we can group them by what they're doing. So we can see 5 pretty distinct clusters of cells and what those cells are doing, what genes are turned on and being expressed or not expressed can vary greatly between the different cell clusters in the microglia, and that can help us know what we should be doing in terms of treatment and how they might be responding. So looking specifically at, what these microglia are doing in disease, we see that in the CLN1 brain and spinal cord, we see an increase in markers of activation or disease-associated markers, and we see a decrease in classical markers or markers that we would expect to be seeing in functional, healthy, more relaxed microglia when compared to healthy brain and spinal cord. And so, for example, a really big gene commonly known for being an marker of disease and activation and linked to other neurodegenerative diseases like Alzheimer's, we see a pretty substantial increase in expression in microglia from these disease samples. But really excitingly, we actually see that expression mitigated by the enzyme replacement therapy. So we're really looking to see how these cell types are responding to therapy. And so now we're also hoping to further analyze what microglia are doing and also see what they're doing in other parts of the system like the bowel. Yeah.
Can everybody hear me? Great. So my lab focuses on vision loss and CLN3 Batten disease, and we are hoping to develop a rational therapy for targeting vision loss. We use multiple different approaches to doing this. One of them, and that would be really helpful in this conference, is if siblings, parents, and affected kids allow us to image their eyes, because what we do is we examine the eye of the patient and look at the retina structure and function, the part of the back of the eye that's affected in the disease, and this provides us important clues into why and what structure is affected early. And especially when we compare it to the sibling, it provides us a proper platform to see, what are the changes? Why is this disease degenerating? What structure is affected? And when we have that information, that allows us to now make a human cell model where we've been able to develop using skin and blood samples from patients and converting them into stem cells. So these are not embryonic stem cells, these are stem cells that are made from blood samples. And then we can create a retina in a dish. And this retina in a dish is a very, very powerful tool to see how the disease is happening and how we can target it in patients' own cells. So if you look at here, this is a control retina, and this is a patient retina, and those green and red dots are basically photoreceptor cells. And you can see, just like in human eyes, these light-sensing photoreceptor cells are degenerated or completely lost. And this is the earliest pathological change that we are seeing when we are imaging patients that have Batten disease. So, as I said, photoreceptor loss, rods and cones. We've used this model now for 4 years to try to understand how this is happening. And I'm not going to go into what we found mechanistically, but we were able to identify a particular enzyme, recombinant acid ceramidase, as a therapeutic for targeting photoreceptor loss in Batten disease or CLN3 Batten disease. And you can see here, again, those green dots are the photoreceptor cells. This is an untreated patient eye, and this is a treated patient eye. You can see the recovery of photoreceptors. And this is now showing both rods and cones. So cones are for the central vision, rods are peripheral vision, night vision, and we are able to basically prevent the degeneration from happening by giving this. And again, so this is an in vitro cell culture model. We've also done preliminary proof of concept studies in a pig model of the disease, a large animal model of the disease, and only treating for 1 week, right? And we try to start before the disease develops here because these are very early studies. And you can see here that within a week's time, the disease progresses so fast. In the untreated pig that has the disease, these rod outer segments are so small and they're degenerating. This is the earliest pathology, the photoreceptor rods getting lost. When we treat for a week, you can see here, these rods are retained better. So long term, we now hope, and you can see the thickness of the retina is already improved, right? It's not degenerating. We're stopping that degeneration from happening, but this is 1 week. We need to do way more to be able to figure this out. And so the future steps are, I want to validate this. Again, as I'm saying, my lab is here trying to image human eyes. If we could do siblings and patients, that would be amazing, and parents, because this will tell us if we are headed the right way, if this is going to work with patients, with people that have multiple different forms of mutations, and whether we are on the right path. And we also want to make more stem cell models and validate it works across mutations. We want to do long-term therapeutic studies in the mini swine model because, as I said, we just had 1 week so far, and we see that. And my ultimate hope is that where we are headed to being able to test this on human patients post-validation. And this project means everything to me, so I would really appreciate, just like to you, any help in figuring out how to do this better. Thank you.
Great work, team. You all got the brief. We're sticking to time. This is great. I think we have Bill to thank. Here we go.
Hi, everyone. I'm Amy Verheil. I am a pediatric nurse practitioner at the University of Rochester Batten Center, and I'm going to talk for a few minutes about natural history research. Okay, so natural history is observational research on something that is living, and we're looking at the course of a life cycle of that thing in their own environment, and there is no experiment here. There's no treatment given. It's important for rare disease natural research to really understand that disease because 1 out of every 200,000 individuals in the US has a rare disease. And we know that even within those individuals, there's a lot of variability. So it's important for us to see as many individuals as possible. We as researchers are interested in understanding traits versus disease markers. So if we see a child and that child is a toe walker and the parents tell us that other people in the family are also toe walkers, we're much more likely to think that's just a familial trait and not necessarily related to the disease. The FDA is interested in how people feel, function, and survive, and natural history definitely gets at all of those as well as quality of life. Retrospective data is looking at things like medical records, chart reviews. We might do an interview related to a past experience, but it's really time-consuming. It takes us a long time to weed through records. And if we're interested in finding something specific, like we want to know when your child walked or when your child spoke their first word, and it's not written in the documentation, then we don't have that available to us. Prospective data is really what's happening at that moment right now with your child. So it's cross-sectional. So on this day, this is how your child is eating, sleeping, walking, talking, etc. If it's collected by an expert, somebody who really understands the disease, sometimes they can notice the difference between participants and also detect some subtle changes. We need high numbers for natural history data. We really have to be able to report outcomes with high numbers. So if we look at 5 individuals and 4 of those individuals lost vision before age 14, but we have 1 who still has vision at age 14, we could confidently say that 80% of children lose their vision before age 14. However, if we have 50 individuals and 1 still has vision at age 14, we can then say 98% of individuals lose vision before age 14. So it's so important to have high numbers. We can identify outliers, people whose story is a little bit different, and that's very important as well. We're really interested in longitudinal data, looking at people over the course of their life and how things unfold, because we know that everybody has a different story, and it's really important to know that. And then ultimately, natural history can be used to compare to a treatment group. And the Hamburg group did a great job with this. They looked at 140 children with CLN2 disease, and that was natural history, and they were able to use that as a comparator for the Brinura trial, which was fantastic. The University of Rochester Batten Center, we've looked at some outcomes as a result of our natural history. We've looked at quality of life, males versus female differences in CLN3 disease. We have validated remote assessment, so we can see individuals all over the world remotely. We've written about behavior, seizures, and we have contributed to clinical guidelines for CLN 1, 2, and 3. The Batten Disease Clinical Research Consortium got formed about a little over a year ago, and it's really exciting. There's 13 different institutions participating, and we are working on identifying commonly collected data, and the goal is to have a larger database, so all of that information we collect from everywhere will go into one large database. So these are my conclusions. Natural history is very important. We really, really ask you to bring your children and participate if possible, so that we can understand as much as possible about all forms of Batten disease. Thank you. All right.
Good morning. My name is Bridget Patterson, and I am super duper not a scientist, but I am a mom of 2 kids who have CLN3 disease. My husband James and I have 4 kids, and our middle 2, Gabriel, who's 20, and Nora, who is 17, have CLN3. And we've been participating in natural history studies at both the University of Rochester and NIH, so I was invited to just share a little bit about our experiences. I'm going to try to go really fast because I'm kind of afraid of Dave. So, like Amy was saying, participating in natural history studies, it really benefits the whole community. Because it's a rare disease, every data point is important. Every participant is really important. And the people conducting these studies will share the data with everyone, with researchers around the world. And so that multiplies your impact, really. And you'll hear from Dr. Dang Do in a couple minutes, and what she has told me is that at NIH in particular, they have the luxury and the privilege of only doing research. They don't have to balance their work with clinical patients, and so they really try to collect a lot of data to help the research now, but also to try to predict what might happen in the future, so that there's data waiting for those new therapies that might come on board. Still, the community is not dealing with the challenges of participating in the study. We are. We know that travel can be really burdensome, especially if you have to fly. And the visit can be really long, like Amy said. So what you see on the screen right now, this is the schedule for my son's most recent visit at NIH at the beginning of June. He had 16 different procedures done over the course of the week. So it was a really long week, and every single one of those involved people asking questions, poking him, prodding him. It was a lot, and we're used to it by now, but I remembered, especially his first few visits, it was really, really overwhelming, and it's really hard. So this is a photo of Gabriel in the recovery room after his MRI a few years ago. And so even though I know how important these MRIs are, seeing him like this is still hard. And getting the reports at the end of the study is hard. Hearing that his brain is changing is hard. Hearing that more cells in his eye are dying, and his retinal nerve is atrophying, so even if there is some brilliant stem cell, replacement therapy that could rebuild the eye in the future, if his retinal nerve isn't working, he still won't be able to see. It's hard. It's really hard. And this is a quote from one of the consent forms pointing out that, like Amy said, you don't get a treatment. You're going through 16 procedures and walking out of there without a treatment. So it's super hard. But the benefits that we've received outweigh the hard things. We consult with doctors who've seen dozens of CLN3 patients. At your local clinics, your local doctor, your kid may be the only CLN3 patient that your doctor will ever see. But these researchers have seen dozens of patients, and they're quick to pass along tips that families share with them. They learn from families, and you learn from them. Everybody learns from each other. It's all a village. It's so valuable. The study doctors are also a resource for us because they're willing to connect with our local doctors and share information that they have that our local doctors don't. So I remember Dr. Mink, who used to be at University of Rochester, he would say, yeah, I talk to these neurologists who've never seen a CLN3 patient, and I tell them, yeah, but you've seen kids with seizures, so treat the seizure, right? They kind of hold the local doctor's hand in a way. They're always willing to do that. And the information and materials that we get from the doctors and researchers, we use those to kind of inform our kids' IEPs when they're in school. And I can tell you from experience that the phrase, we've been working with doctors at NIH and the University of Rochester, and they recommend XYZ, can carry a lot of weight, which is really great. The most important reason and why we will continue to participate in natural history studies as long as we possibly can is that they provide my kids an enormous sense of purpose and pride. They know that they are doing something that no one else can do. So Gabriel was subject number 1 in the NIH study. And he just completed his 9th visit there. And Nora has been to 4 visits at NIH. And we make sure that they know how special they are. And it keeps us all going. So, sorry. So I'm going to put this slide up. If anyone wants to contact folks about natural history studies, I know that Dr. Dang Do and Keisha at NIH would be happy to talk with you. I know that Amy would be happy to talk with you. If you have any questions for me, I'm certainly happy to talk with you anytime this weekend or anytime after this weekend. But that's what we've experienced as part of natural history studies. So, thank you.
Bridget, thank you so much. We see how challenging that is to share, but I'm lost for words at how important these natural history studies are, and thank you for sharing such a raw account of how challenging it is for you to do that. But despite that, the benefits, and you should feel proud, and your kids should feel proud, and we owe a debt of gratitude to families just like yours that do this to help us move that needle forward with research. So thank you for agreeing to be here. I know we put you in the hard spot, but Dave didn't crash tackle you, he just wanted to hug you. Doesn't happen often.
Well, good morning. My name is Melissa Pratt, and I come from Jill Weimer's lab at Sanford Research in South Dakota. Today, I'm going to be talking a little bit about our work identifying blood-based biomarkers as a part of a larger effort to develop signatures for Batten disease. Just to start off with a quick acknowledging of all the folks in our lab working hard every day. So briefly, our lab approaches Batten disease research as a connected process. We work to understand what's happening at the micro level within cells and in the body in Batten. We develop tools such as animal models to aid in these studies and to test therapies that we develop. Responses to those therapies then go back and inform things about the disease so that we can further refine our approaches. And all of these pieces work together to support disease understanding and therapy development to move those therapies forward. A key part of this, highlighted here, is discovering new biomarkers, and that's my focus here today. Biomarkers are measurable indicators of biological process, condition, or disease, such as a protein or a molecule found in the blood. Cholesterol would be a common example for a biomarker for heart disease. Current methods have a lot of utility, but they have limitations. For example, this CLN3 scale that you see here, there's a general trend that scores get higher as kids get older, but there's a lot of variability within time points and in how those symptoms are measured and reported, making it really challenging to track small but important changes, and can also make it really difficult to see whether a treatment is working and almost impossible to measure treatment response over a really short period of time. So this is why we're working on discovering blood-based biomarkers to add clear, measurable data linked more closely to the biological processes that are happening in the disease. That will just help track progression and more precisely work in combination to strengthen those metrics and gain a more accurate, quantifiable picture. So a couple examples of these blood-based biomarkers are GPDs and neurofilament light. I'm not going to get into the weeds for the sake of time, but our lab identified GPDs as being changed in patients with CLN3, and neurofilament light you'll recognize is currently used in a number of trials, currently tracking efficacy for people with CLN2 on Brineura, for example. These individual markers are useful by themselves. They still lack the ability to more broadly inform the status of disease progression at any given point. So that's why we are working to combine multiple blood-based biomarkers such as these into a broader biosignature score that can be combined with other clinical metrics like vision, gait testing, ultimately just to better track disease stage and treatment response. So currently we are in the discovery phase of this process. We have blood-based readouts for our pig models of CLN2 and CLN3, and thanks to support from the BDSRA, we are able to work in collaboration with Tom Wishart and Nadia Mitchell to get those same readouts from their CLN1, 5, and 6 sheep. We are looking for markers that differ between animals that have the disease and that don't, and at the same time we're also screening human samples to see which of those markers, hopefully many of them, are also changed between healthy individuals and those living with Batten. So to summarize, we are looking at blood-based samples from animal models and humans, and from various forms of Batten disease. We're looking at all forms to find as many things that change biologically in response to the disease. We're then going to use some fancy modeling processes to combine all of those metrics with clinical metrics to form disease-specific scores. And the goal is to have markers that are translatable to humans and meaningful for tracking disease and therapy response. Thank you.
And now continuing on the theme of biomarker studies and natural history studies that Bridget and Amy were just talking about at the NIH, we have Dr. An Dung Do online. So I'm going to hand over the driving seat. Yep, we're good to go. Hi, Anne.
Only speaker who can claim that I am completely immune from being physically tackled, but I'll try to keep to time. Next slide, please. What I'd like to share today is our...
Got it.
Sorry, can you hear me?
Yep, we can.
Oh, okay.
Sorry, I'm just going to advance the slides.
So what I'd like to share with you today is our efforts to contribute to the CLN3 disease research field. As Melissa had just described to you, and you might have heard recently in the news and throughout this conference, there are multiple promising candidate therapies that are in the pipeline. And one of the major aspects contributing to moving these research projects forward is to have multiple options to evaluate how the disease progress or how a potential therapeutic treatment would be efficacious or not. And this is considered biomarkers. Next slide, please. How we've been working on this is we started a natural history study of individual with CLN3 in 2017 and been having a wonderful privilege and opportunity to meet families and children from the US and throughout the world. Next slide, please. In the natural history study, they come for, as you heard previously, a quite extensive evaluation that include clinic-based evaluations such as neurodevelopmental testing, as well as tests and procedure including brain imaging. The MRS that you see there stands for magnetic resonance spectroscopy, looking at certain metabolites in certain areas of the brain. We also collect concurrent biospecimen because CLN3 is a neurological condition. We are interested in looking at the cerebrospinal fluid. That's the closest that we can get to studying what's going on in the brain. Next slide, please. And up to now, we've been able to evaluate 47 individuals, which is what's shown in this quite complicated graph. About 2/3 of those individuals have come back for annual visit, either in person in the blue dots or remotely in the red triangles. And what you see there in the blue line and the red line are the median age across these visits of the study participants. Next slide, please. So I'd like to give a concrete example of how we use and try to incorporate the clinical imaging and biosamples from the natural history study. Melissa had briefly mentioned this protein called neurofilament light chain, which is something that is produced in the brain cells upon damaging of the brain cells by any cause, it seems. This NEFL would get released into the CSF, cerebrospinal fluid. A small percentage of that will also get secreted into the blood. Next slide, please. So what we've done is we looked at the CSF that we collect in the natural history study, and we've described and reported that in individuals with CLN3, the level of NEFL in CSF is higher compared to individuals who do not have CLN3. Next slide, please. Incorporating this with the clinical data, we show that there's a correlation of higher NEFL level in the CSF with higher disease rating scale using the University of Rochester Unified Batten Disease Rating Scale. Next slide, please. The higher level of neurofilament light chain correlate in the reverse direction with neurodevelopment skill. So the higher level, the lower the performance on neurodevelopmental tests, as well as lower level of brain metabolite that we quantify from MRS data. Next slide, please. How do we apply this in real life or in real cases? So the Beyond Batten Disease Foundation and Theranexis have been working on a candidate therapy called Batten 1. And what they've shown, next slide please, in a small safety phase 1 study is initially we see this high level of neurofilament light chain in the CSF and blood. Next slide please. And after 18 months of the individuals on Batten 1 therapy, both the level of NEFL in CSF and blood decreased. Next slide, please. So to reiterate and echo what have been spoken previously, and probably you'll keep hearing throughout this conference, are that natural history is a very important part for us to be able to study these rare conditions. It is effort and resource intensive, but if we use thoughtfully the data collected and do good science, it will provide fundamental information for generating and testing hypotheses, such as the NEFL biomarkers and others that are listed here. And also provide samples so that other basic science researchers will have the opportunity to collaborate with us. Next slide, please. It goes without saying that this natural history study cannot be done without the participation of the children, the families, the siblings, the parents of individuals with CLN3 and of the support and advocacy groups listed here. Next slide, please. And since this is a huge endeavor, as you have all heard, everyone needs to be involved. And these are some of the collaborators that we've had the privilege and the joy of working with and looking forward to continue our collaborations. Thank you for your attention.
Thank you, Anne. And if you're able to stay on for the Q&A later, we'd love to see you back or hear you back.
Okay, so I'm Paul Trippier, I guess local boy down the road in Omaha at the University of Nebraska Medical Center. So I am a small molecule drug discovery guy, so you're going to hear a little chemistry. We are very active in looking for small molecules that can modulate proteins in diseases, CLN3, CLN6. And we're active down here. We look for targets, we look for compounds we can modify to make better drugs and to become drugs, removing negative qualities like toxicity, and then we move forward with preclinical studies in animal models and stem cells to push, hopefully, through to clinical trials. The work we do, we're about at least 10 years away from actually getting something into the clinic, but we're at the start point. So brace yourself for this next slide. This will be on Coach Bill's quiz, so start memorizing. So our starting point is this flupertine compound. Many of you would have heard of this. There's some controversy around it being potentially effective, or at least having a protective effect in CLN3 disease. The problem with it, it is highly toxic. It leads to liver toxicity. We know why. It forms this compound here, and then highly reacts with other proteins and itself to induce liver toxicity. So never going to be a treatment option for CLN3 disease. Previously, we've taken this compound and we've played around with it, making changes on the left and right-hand side, and we've managed to reduce the toxicity and keep the protective effect. What I want to talk about today is a new compound where we have completely ameliorated the toxicity and kept the neuroprotective effect. So before I do that, that first generation compound where we've added a modification on this right-hand side, this work is with Rosie Bustani out at the American University of Beirut Medical Center, where she has CLN6 mice colony, and treatment with this compound has an effect to rescue several characteristics in CLN6 disease. We can improve motor coordination, we can remove storage material from the retina of male and female mice, we can reduce cell death in the brain, and we can protect neuron numbers in the brain. So very excited about that compound going forward, but there is a toxicity issue with it. The new series of compounds we've previously published using stem cells taken from a CLN3 patient that we've been able to drive out into neurons. That's an excellent model system for this disease, and we've now been able to convert that into an assay format where we can screen several compounds at any one time. So if you take a look at this very big long bar graph, flupertine here, that starting compound, has a little bit of protective effect, nothing really to write home about. Our compound I just showed has significantly more protective effect, and then along with a whole bunch of compounds that don't do anything, we have managed to identify several compounds which have significantly more protective effect. Those are the compounds. Don't expect you to take this home, but compound on the left, 20% protective effect. This compound, 74% protective effect, so it has a significantly better effect than flupertine. We also show that these compounds, in line with the protective effect, also get rid of SCMAS in this cell line. So looking forward to pushing this forward and developing these. Here's the group down the road that do this work, and we're very happy and thankful with the NIH for funding this. Thank you very much.
Thank you, Paul. Down to our final three speakers for this session.
All right. Hi, everybody. My name's Eve. I'm a research specialist at the Cognitive Neurophysiology Lab at the University of Rochester Medical Center, and I'll be talking about auditory processing in Batten disease. So first, why are we studying auditory processing? You've heard a lot about most other sensory systems, but not too much about ears and auditory processing. So we know that individuals with Batten disease have progressive loss of cognitive functioning, speech, vision, and motor skills, which makes it really hard to do the work to accurately assess their neurocognitive skills. And you've also heard a lot about how we need to find a biomarker, which is a quantitative measure of, specifically for us, brain functioning that can be tracked fully across disease progression. And auditory processing appears to be pretty intact during the progression of Batten disease, so we think that measuring auditory processing across disease progression might be pretty useful at measuring the integrity of central cortical processing, so what the brain is doing. And we hope that this will eventually serve as a marker of disease severity and treatment outcomes. So how do we study this? We use electroencephalography, or EEG for short. I know that a lot of you have terrible experiences with the clinical version of EEG. I promise we use this version, which is a funny looking swim cap situation with a bunch of gel, which washes out just like hair gel. Way less invasive, no glue, no tape. And we use this because it's a non-invasive way to measure electrical activity at the scalp that's produced by neurons firing in the brain. And we can use EEG to measure when and to what degree your brain responds to certain stimuli even without paying active attention. And we use event-related potential recordings to allow us to track your brain's response immediately following, in this case, an auditory stimulus, so a beep, a boop, or a word. We study this specifically in two ways. The auditory mismatch negativity is a recognition of deviation from a pattern, so we're looking into sensory memory, and we're using EEG to detect the different sensitivity of your brain to frequent versus rare stimulation. And we expect that the brain responds more to rare stimuli, so if you hear like beep, 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 boop, like down here, you'd respond more to the boop than you would to the beep. You kind of go like almost nose blind, but with your ears and your brain, you process less of the bad smell once you've been in a room for a while, where somebody who just walks into the room might go, whoa, that's a terrible smell. And that's how you respond to the boop, and this does not depend on any attention. And we have some previous work from our lab that I don't have up on the slides here, from Dr. Tupi Brima, that shows that there are differences in the brain's response to the deviant stimulus in specifically CLN3 disease. We're also studying semantic processing, which is how your brain processes language using an ERP called the END400, which is a component that's considered an index of semantic integration, so how well your brain is processing language and how your brain is processing language. And we expect to see a larger response to unexpected words. So if I were to say something really wacky, you'd process it more. You'd be like, why is she saying that? Instead of, you expected to hear me say. And so we study this by having participants listen to sentences that either end in a congruent or expected word or an incongruent word, and there's no response expected. So if I said the sentence, I baked a birthday, you would expect me to say cake, which would be the expected ending, and you wouldn't really process that. You wouldn't think too much about it. It sounds totally normal. But if I said, I baked a birthday clue, you'd be a little confused by what I was saying, and you would think a little bit more about it, so that's where you would see that larger response. And then some future applications of our work is that we hope that these auditory ERP measures may serve as helpful outcomes and measures of disease severity and treatment response. And we hope that it can be used as a measure of success in clinical trials, which similar work from our lab is currently showing promise as an outcome measure in trials for Rett syndrome. We're doing this research at the conference. We're just at the table outside if you want to talk to us. We're doing the EEGs right upstairs. And then a huge thanks to everybody listed here from my lab, from the Batten Center, and all of our participants and their families.
Who needs a gym when you can just jump up and down on the stage multiple times? So when you come out, when you finish this one, you just close the link, close it down, and then double click on the next one.
Okay.
Is that okay?
Yeah, I think it will be.
And then you just hit presentation mode, and you're off.
Great, thank you. Hi, everybody. I'm Jen Vermillion from the University of Rochester, where I direct our Batten Center, and I recently took over as chair of our BDSRA Centers of Excellence program. I'm going to do something a little different here, where we're going to talk kind of at a higher view of things and not a specific research program, but thinking more about collaboration and how we're trying to advance that. So we've had 1 year of our Centers of Excellence program. This was launched in 2024. The initial chair was John Mink, who more recently stepped down. This was developed by the Batten Disease Centers of Excellence Consortium that had leading Batten disease clinicians. There was parent advocate on that program as well, and then BDSRA Foundation representatives.
And together they determined what is the mission of a Centers of Excellence program. They used national model and standards for designation as well. And from all that came up with a mission that I won't read through here because we have that 4-minute deadline, but just to give you a sense that we came up and said, okay, what is the mission of this program and what are the main pillars that makes you a center of excellence? So the main things are optimal care, enhancing access to care, how do we advance research, and how do we improve education and collaboration. So from the first year, there were 5 centers designated as well as 4 affiliates. So what we had was you're a center of excellence if you meet a certain number of standards that are set forth, and I've listed here in the teal the 5 that were designated in that first year. And then there were a number of centers who are doing amazing and excellent work as well, and we hope to, through some working together, bring them to a center of excellence designation as well. And so I've listed the 4 centers here who've been doing great work. So what have we done in this 1 year? We started off with a bang. We've been meeting very frequently. Our group meets every 2 weeks where we talk either about more research-focused things, clinical issues, advocacy. We have speakers who come from other groups as well to present and talk when we're thinking about how do we move forward a certain idea. We discuss our clinical programs. What do we need to do to move our clinical programs forward to provide the best care for patients? And then we discuss our research priorities and scholarly activities. How can we collaborate on putting out materials to better understand the different Batten diseases? How can we advance natural history studies? How do we come up with treatments? All of that we try to discuss in these activities. From this, we've really advanced our collaborative research. We have developed the BDSRA, which is where we're really trying to bring forward clinical research and develop a way to collect natural history data across the sites. We're trying to improve our widespread NCL knowledge. So that is where we're also trying to go out into the world and say, okay, can we put forth manuscripts where people understand more about Batten disease? We have a gene reviews overview of the NCLs that's gone out, and that is something that a number of clinicians will use and go to is this resource for when they have a patient with a disease they don't know as much about. They'll use gene reviews, and now we have an updated version out there. And then we're trying to improve patient access and care as well. We are trying to grow and expand. We have new applications open for those who want to join the program, who want to come together and provide excellent care for patients with Batten diseases. And so those are now open for people to come in as an affiliate status. And then after working together for at least a year, they have the opportunity to apply for a Center of Excellence designation. And then the prior affiliate programs that we've worked with who've been doing amazing work, they now have the opportunity to obtain a Center of Excellence designation as well. So this is information for if you have any group that you work with or local clinicians who you think want to be part of the affiliate or Center of Designation program, they can reach out to the COE at bdsrafoundation.org to find out more about the application. Okay, now I'm going to switch over to my next one. Okay, so now what I'm going to do is I'm going to talk about a conference that we put on more recently that was about innovative Batten disease clinical trial design. And this was a multi-stakeholder conference that I'll talk about. So this was put on through a program called NeuroNext, or the Network for Excellence in Neuroscience Clinical Trials. This is an NIH-funded network where the main goal is to have multiple sites across the country that have trials that run through them, where we have more efficiency in the clinical trials, where we're able to test new promising therapies in different neurological disorders, and then respond quickly as opportunities arise to test more promising new treatments. And so if we have this center where it's able to move things through more quickly with NeuroNext. The Network Linked Open Conference, which is how this conference came to be, or the NLOC, is an opportunity that was put together by the NeuroNext group, where we take resources from NeuroNext to really focus on an area of unmet need within the research community in neurology. And this was the second year that this conference had been put on, and different people put in applications for different topics, and Batten disease was the one that was selected this year. So what we did for the conference this year is we focused on something called Master Protocols for Rare Diseases, with really thinking about it as a framework for Batten disease. So what are master protocols? These are trial designs that use one main protocol across multiple studies, so that you can just feed studies into this one design where there's shared infrastructure, you can adapt to real-time data that comes through, and the goal is to really accelerate the timeline from when a drug comes into a trial to where you figure out, does it work or not, can we move it toward potential approval? So when you think about master protocols, there are different kinds of designs within that can come out. So there's a couple that we really focused on for this conference. One is called Umbrella. That's where you have a single disease and then multiple treatments where you say, okay, we're gonna have people either get treatment A, treatment B, treatment C, and see how they work. Or there's a platform trial where you have multiple diseases, think about it like CLN 1, 2, 3, etc. And you have a single treatment where you try it in all those different diseases. And so we really focused on what are these, what do they mean, what would it look like to have either of these set up for the Batten diseases. This conference where we talked about these topics was really well attended. We had 226 people who registered and 185 who attended. We had a number of people from the Batten science community, but we had a lot of people from outside of it as well, which really brought fresh ideas and perspectives. We had clinicians, we had research team members, people from the NeuroNext network, industry representatives, and patient advocacy representatives. And from all of this, we talked about how this would look for the Batten diseases. And the next steps we have is, okay, taking the information we learned, how do we set up a clinical trial that would really be able to move treatments forward in the Batten diseases? So right now what we're doing is we're trying to set up meetings with relevant foundation and industry collaborators to map out a path forward from the information we learned from this. We're also working on setting up a timeline for engaging the FDA and putting together a master protocol trial for the Batten diseases. And then from everything that we learned from putting this together, we're also working on developing a manuscript where we can talk about, okay, what did we learn about how we put together master protocols in rare neurological diseases? And that's all.
Good morning. My name is Jenna Soper. I am the Executive Director at Lattice Bio and the lead for our CLN2 program. And the drug candidate that we're developing is still in the preclinical stage. So today I'm going to be showing you some data from our preclinical studies, our animal studies. And I have to disclose I'm an employee of Lattice Bio. Okay, so we have heard a little bit today about the basics of gene therapy, and at Lattice we believe in the gene therapy approach. We also believe in enzyme replacement, like Brineura, which we've heard a little bit about. So while Brineura can deliver the TPP1 enzyme to the CSF, you can see that after a few days, the levels drop back down to below baseline. We believe that a one-time gene therapy could actually result in high levels of TPP1 in the CSF for a steady state, for an extended period of time. And we believe that this treatment could actually be more efficacious than Brineura and could alleviate some of that treatment burden of repeated long hospital visits, because it would be a one-time treatment. So to test that hypothesis, first we looked into the mice, and we used a transgenic mouse model of CLN2. And we injected our drug candidate, LTS-101, directly into the ventricles of the brain. And you can see here on the left
that LTS 101 resulted in a very significant increase in TPP1 activity in the brain, over 100 times more than when we injected just a vehicle or like a placebo alone. It also significantly reduced the lipofuscin protein. That's the lysosomal storage protein that gets abnormally accumulated. So you can see on the right side in blue, significant reduction in that protein as well in the brain. We also looked at the effects of phenotype of the CLN2 mice. And you can see on the left that LTS 101 significantly rescued the tremor phenotype of these mice, so reduced it almost back down to the level of the healthy carrier. And it significantly extended the lifespan of these mice by about 50%. So after these really successful studies in mice, we moved on to non-human primates, or monkeys. And we did the same thing. We injected LTS 101 directly into the ventricles of the brain. And you can see here that we saw a very high level of TPP1 protein in the CSF. So it's this blue line with the blue squares. Over 5 times the level of just the baseline. And importantly, it's higher than the level in the gray bar, which is about the range we estimate that healthy humans have. So we were really excited to see that this could potentially be a therapeutically relevant level of CSF TPP1. Now, very importantly, we also tested the safety of LTS 101, and we're happy to see that it demonstrated a very good safety profile in the non-human primates. It was well tolerated, and we had no adverse events or toxicity. Based on all of these data, that the fact that it increases survival in mice, that it reverses the tremor phenotype, that it's safe and tolerable in non-human primates, and that it produces this potentially therapeutic level of TBP1 in the CSF. And one piece that I didn't get to show you, but that we have high biodistribution only to the brain. It does not look like it's kind of escaping into the rest of the body, into the liver, where we might see some toxicity. So with all that data together, we were really excited and think that LTS 101 is a promising gene therapy candidate to treat CLN2. And we are working to develop it for the clinic. So it would be a one-time injection into the brain in just like a one-time neurosurgical procedure. Thank you very much.
Take a seat. Thank you so much, Jenna. If you were one of our speakers, would you like to come and join us up on the stage now? I have to say, we started 20 minutes late and we have finished bang on 1 hour, so well done, guys. That was great drills. And now we have time for 15 minutes of Q&A. There are 2 microphones to either side here. You're welcome to line up behind those. And we have at least 1 microphone, yeah, anywhere you like. And we are also, so we will have a break after the Q&A, but we have deliberately made this session right before a very long lunch break in the full expectation that we'll be running on Batten time and probably be running over. So if you are a speaker in the next session or somebody that really wants to hear those presentations in the next session, we will have ample time as well. So just to take down the anxiety, we have ample time. So do we have anyone that wants to, oh, sorry. Okay, so speak into the microphone when you are answering questions, please. Are we missing somebody? Dave, you're supposed to... you can sit up here. We have an extra chair. It's not the same without you. So, we have a question here? Yes? Excellent.
Yes, thank you. So we also have in the audience our clinical operations lead, Seuss, and she's going to give a little update about that in the next session. But yes, we are moving as fast as we can towards the clinic and doing everything we can. We're working really hard at the preclinical data that I showed today is wrapping up, so we are moving as fast as we can, absolutely.
So after that, will you be looking for patients for that? Okay.
Of course, yes. We're going to be thinking about putting together a clinical trial. We're thinking about putting together, which sites where we're going to be located, and we're putting all that together. It's really premature. It's hard to predict the timing just based on FDA hasn't, reviewed our package yet. So we're kind of at the mercy of some of those things outside our control, but we're working as hard as we can. We will be looking for CLN2 patients in the future, yeah.
Okay, thank you.
Thank you. We are, yes, so the gene therapy I showed today would be instead of Brineura. So if the patient has been on Brineura, that's fine. We would, still be able to treat that patient. And the idea is that, if the gene therapy doesn't look like it's working, we can go back and resume Brineura. So that would be in the future. We're still developing our clinical protocol. We're still developing on which patients we'd be able to start with first, but we are not trying to limit at all, your exposure to Brineura, and, we can, it wouldn't be exclusive of Brineura.
Sorry, so if they did do the clinical trial, if it went through, then they would stop Brineura after the one-time gene therapy?
Well, that would be the idea. I mean, the idea of the gene therapy is that you would have a one-time treatment. It would be a neurosurgical procedure, so longer, more involved than just the infusion, but then that would be it. You would be on that for the lifetime without having to go back and do repeated Brineura, so there just wouldn't be the need. I'm just saying that if it turns out that for whatever reason you want to go back on Brineura, it shouldn't be exclusive.
Thank you.
And I can see our clinical ops for Lattice Bio is also presenting in the next session too. So it'll be, great. There'll be lots more questions, I'm sure. Are there any others? Yes, would you like to come up to the microphone? And if you do have questions, please feel free to queue at any time. You don't have to wait your turn. You can just stand behind in the line.
Hello. So my daughter has CLN6, and I've connected with many families also who have CLN6. And what I found is a lot of our children are not in the US. They're in other countries. And it's created a major barrier to just having these children and their information involved, even in like the natural history studies. I know the families want to be involved. And I want to know how we can overcome that barrier, because even in the hospital I work in, or just some of the families I've connected with, there's people that can translate, that can do all that, and how can we like bridge that gap and make it happen, because there's so many of these kids that are they're not on the radar.
In terms of connecting, so we have patient advocacy groups that are working together closely to try and bridge those gaps as well. I think this is probably a better question answered for the sites that do the natural history studies, because they do have international offices and also off-site, like telehealth opportunities depending on the study. So,
yeah, and for our study at the University of Rochester, we can do remote visits as well, and so we've had people participate from other countries in that way. Our study is currently an unfunded study, so we don't have funding to be able to pay for translators, but if somebody's available and able to do it, we're happy to work with them in that way.
So when we do, and another problem I think is with CLN6, I've even experienced it here, is getting the connection with our providers to the experts. It's even harder when they're in another country. I found so far a lot of the providers do speak English. Is there an ideal way to connect, to make the connection with somebody so that, you know, they can advocate for the families to be included and also be up to date on, you know, all the information?
Yeah, so at least for our center, we have an email address, and so we've had clinicians from other states or countries reach out to us that way, and we can provide information in that format. And if it's just kind of back and forth email, it can be more general without patient information. And if they want to do something more official, we just need a release from the family to say we can speak about that. Otherwise, we can keep it more general if we don't have that information.
Okay, thank you. And you'll find most of our centres of excellence have that capacity as well. So Kennedy Krieger, for example, is an international office. I know Emily Dellis Reyes is here. We're from Nationwide Children's, does international telehealth as well. So going through those channels and reaching out to the clinic directors, or if you go to the website for the centres of excellence, they have their contact points there. And so most of them do have that availability and capacity as well.
Okay.
Yeah.
My grandson has he's CLN2, and I noticed a lot of the research was on 1 and 3 and up. How much crossover between the different types is there, and how much value do you share between the different types?
So we don't have any favorites. We look at as many as possible, 1, 2, and 3, and actually all those, many of the things I talked about were CLN2, things we're doing with seizures in mice, things doing with the bowel in mice, things to do with peripheral nerves. The answer is all these diseases are kind of similar, but they're also kind of different. They're similar enough that they've been grouped together as one group, but it's a different protein, a different thing inside the cell that's gone wrong, so we might expect there to be some differences in how the brain is affected. And we find some quite big differences, but when it comes to therapy, the main frontline approaches for enzyme deficient forms, so CLN1 and CLN2, are either giving back what's missing, that's what's happening with Brineura, or gene therapy like we've been doing, or like we've heard from Lattice Bio. And that has to have another very specific tool to do it. The main approach is the same, giving a functional copy of the gene back, but the actual tool that you use to do it is specific to that form. Now CLN3 is a bit different again, because it's a whole different category of thing that has gone wrong. And again, some of those approaches might not be applicable for it. Does that answer?
Yeah, I also have a question about, my grandson had an infusion, you know, he does it every 2 weeks, and they did it 2 days early this week because they were coming to the conference, and it was like night and day after the infusion he had 2 days earlier. He was like unbelievable on how alert he was, how smiley he was. And so my daughter wonders if she should be fighting for trying to get those things compressed a little bit, or is it because from the chart where it's up and down, is he just not so down and up again? And what do you think about the infusion being 2 weeks apart?
I think it's a really interesting and helpful observation to have that. I think it's hard to say because there can be that variability. Perhaps it is helpful to have a shorter time frame, but I don't think we know enough yet to fight for it because we don't have the information behind it. And the other thing we don't know is there any problem that could happen if we did it more closely together? So, but these observations are helpful to collect because that fuels people to study it to better understand if that's the right thing to do.
All right, thank you very much.
Hello, so my name is Kelly and I have my daughter Serena, which is CLN3, but my question is not about her exactly, it's with the biomarkers. The biomarkers that we're seeing on the blood and we're developing right now, can in the future be something done to see like where somebody is pregnant and they try to find out like they just go for Down syndrome or like any other disease? Can we figure it out to give this as a kind of a part of that testing to be added So the kids will be like, okay, we know that we have somebody like coming in life with CLN3, CLN2, even the Batten's disease.
Yeah, so there's a few factors there. The first one being newborn screening panels. We've all heard of them. There's a limitation to what can be added to the newborn screening panel. It has to have an approved therapy. And so those things kind of tie together. We also need to know early biomarkers that are changed very early on in order to get something that we can detect right away in that panel. There's also genetic testing.
Which is like more
that field's advancing very quickly. Being able to ultimately know before they're born would be ideal, but there's risks associated with that too. So the answer to the question is yes, there is interest and development happening for prenatal and early neonatal
because it will help us a lot of our community with the families that want to expand, but then we are always scared that the next kid that will come will have the disease or not. So that's why I'm asking. Another question is like...
Before you ask the next one, I will add to that too, that we have a gene panel session this afternoon, and we have 2 genetic counselors and a medical geneticist that you will... This perfect question for that panel as well.
My next question is this, because like when my daughter got diagnosed with CLN3, I had so much hope, because on that time, Dr. Emily with Ohio group, she was developing the gene therapy. I want like somebody, one of you guys, to explain to the new families that how we do the research and whose kids will be qualified. Because this is a heartbreaking for us, where I learned how you choose to get those kids about 4 years later. And I can explain how my doctor explained to me. So my doctor said, like where we develop a research, we want to have the ones that gonna have like, they are newborns, that they have that disease. They are like, so they will have longer time to live compared to the other kids, which my kid was 8 years old on the time, and I had so much hope that she will go on the trial. She couldn't make it the trial because like, as I understand it later on, that was like kind of like, we need to collect more data, we need to do the development of the treatment, we need more life expectancy. So I need that to be like known to the other families that they are new with kids with, they are just like diagnosed now. Because like, I know that the researchers are hard, but it will be like better to explain to them like, okay, your child is like 10 years old. And if we go like, so that will be a good idea to explain to the new families.
I'll go first, and then you. Or
Yeah,
you
no,
can,
you can.
I'll go first because I can be neutral on this. So I mean, so every clinical trial initially is what we call, it's going to be the first time it's ever been done because of the rarity of these diseases. So the most important criteria is always safety. So that's always going to be an evaluation for the children in terms of, is it going to be safe to administer this? I know that's, you know, this is maybe the hope that you want.
Which was like, my understanding was the good thing that you don't have the placebo group on those researches. So there is only one group that will be seen and not compared to another group where we give the placebo.
Depends
and
on,
we get
it
like
depends on the
so
intervention.
this gets into, you know, the types of genetic therapies. We're talking about gene therapies. This is what they call it, an open label type of thing. They don't have placebo controls because, and the FDA has different rules. This could go on for a long time. I'm going to try and be clear here. We got this in discussion last year, so I'll use examples. The gene therapy is usually no placebo control. I'm going to get into the selection criteria, which was your question, but like the miglustat does have a placebo control, and that's simply because there's two arms of the FDA. One does biologics and one does drugs, and they are not aligned in terms of that.
Got it.
So that is something that is a bigger picture discussion in terms of things. But to answer your question and how you select, so the patients, it's the companies that own this that put the criteria in.
Okay.
They do the selection, and they remember these are, and I'm just have to say it, these are companies that are for profits, so they want as much possible success, so they will pick the children where you're going to have the most likelihood of success to start with, okay?
Yes.
I'm not going to look at any of the companies in the room because it's not fair, but
No.
and that's dead note. You heard me ask what's the inclusion criteria for latest, right?
Yes.
Because that's going to be very important with respect to it. Different companies are going to design their trials very differently based on their expectations for a variety of different things because they have a lot of data that they know what's going to be the best thing. So for the new families, the criteria is just ask a bazillion questions
Yes.
and advocate and find a physician to advocate for you and use the BDSRA to advocate for you.
What is like hurting on that time where I got it, the thing though, they gave me so much hope, or like the genetic doctor gave me so much hope that my daughter was going on the trial that I was like amazed by that. And then I had the nurse reach out to me and says like, hey, as of right now, we're still looking and we're still putting kids on the trial, but there are more trials out there that you can reach out, where me and her knew in the same time that they weren't anything. So that is like a devastating news to a mom that is like, okay, you're putting me up in heaven and then you're dropping me. So.
Yeah, I think, I suspect you're echoing a lot of what a number of people here have experienced, and there is a period of time, and probably around this time, where there was so much going on in the field of therapeutics, and then everything sort of seemed to fold or close for different reasons. And I think it was devastating for everybody involved, and it's one of the challenges that's come up along the way for multiple rare diseases, but for many of the Batten diseases too. And so I think that's a really good point to bring up, and it reminds us also as clinicians and researchers to temper expectations, because we don't always know and we don't have control over it either, but it impacts you and your family the most, of course.
And like the major thing is like they will tell you like, oh, just go on the website and check.
Yeah.
Just go on the website and check. You check that website 10 times more than I do as a family, because I do not have that knowledge.
Yeah.
But don't put that hope up for me that will be like, I'm like over the moon, and the next week I'm crying and screaming in the corner saying like, but why not my kid?
Yeah.
And this,
But
I think it's a
thank you.
really, really important thing to raise, because we have all of our industry partners here with us, and I know they already deeply empathise with the system, with the situation, but then they also have systems with the FDA, and as Jen was saying, there are a lot of other variables that we and they don't have control over, and it is just one of those horrendous situations where families like you have to deal with this, and
I think it's mostly the American way to say no, where you sugarcoat everything and then you serve it.
Yeah.
Because like European style is like, I tell you right away, no.
Yes.
But thank you.
Well, thank you for your comments. It's
Okay,
very
thank
helpful.
you. Thank you so much. We've got time for one more question, and then we'll have a short
Yep,
break.
I'll be pretty brief. So my name's Ned Timmerman. I am the father of Jack, who is CLN1. My question is for Dr. Cooper, because you mentioned systemic gene therapy to help as opposed to just central nervous system gene therapy. What is your thought on how to combine those, and what would sequencing that look like if you're combining central nervous system with systemic therapies?
Very good question. So obviously all our attention has been on the brain, but our work is suggesting that, well, treating the brain does a lot of good things, but if you don't treat the body as well, you won't get as good an outcome. So when we're doing this, and remember we're doing this in our animal models, we're doing this in mice, which are very small and very simple. We give that as early as possible, within a couple of days after the mouse has been born, and there are different degrees of difficulty. When we inject into the bloodstream of a mouse injecting into a young mouse, you can imagine its bloodstream is tiny. It's not easy to do. We have some really smart people in the lab, much smarter than me, who can do these things. And we do that first because it's tricky, and then we do the brain treatment, which is actually quite easy. That's not such a tricky thing. So we sequence them in that way in an animal model. Obviously, that's a mouse. We also have sheep. Our sheep are much bigger, they're more complicated, and we made them specifically because their brains are more like a child's brain. They're much bigger, and we can address practical problems of where does it go. We heard from Lattice about where does their gene therapy go. We asked the same questions in a bigger brain. So we will do, and we are now doing, treatments where we do both brain and body and see how it goes. And does it actually work? Our main goal is to know, is it going to do any good? Are there any problems associated with this? And there are some problems that are associated with doing gene therapy intravenously, which may mean we have to come up with some better tools for doing it, which we're working on. So that's going to take us some time. We want to be sure that we've got that working well in one of our systems before it would ever go forwards to doing something else. And then we'd have to face another set of regulatory things about getting approval for delivering gene therapy by multiple routes, which would then add another layer of complication on top of that. So what we're doing is theoretical work, trying to know, will it work? We will get that answer in animals, and we will work then to see how we take that forwards.
Thank you.
Thank you.
Happy to discuss later.
Yes, and thank you all. If you would join me in thanking our speakers, please feel free. Yeah. Please feel free, they're all very friendly faces, as you can see, to go and approach any of the speakers this weekend. That's what they're here for. You've now speed dated each of them, so pick your favorites and go and introduce yourself. Thank you all. We'll take a 5-minute break. Are we okay with a 5-minute bio break? Is that going to be okay? Or put your hand up if you'd need longer than 5 minutes for a coffee or a quick bio. Okay, let's be back here in 5 minutes' time. Stretch your legs and we'll kick on with the clinical trials. Okay, guys. Well, Noah is rounding people up outside. I really just wanted to give a very quick overview of the clinical trial for sort of a basic 101 so that we can have a little bit of context for those that might not be so familiar with how the research and development flows from molecule discovery right through to regulatory approval for a therapy. So essentially, we've already heard a lot from this preclinical phase. So the preclinical is pre-human studies, and that involves some of the things you've heard about today. So be that in sort of the cell culture models or the test tube models, if you like, into small animals and into larger animal models. This is really that preclinical phase where this discovery and development takes place. Once we get into the human studies, so that is when, so in the US we call this an IND, is given by the FDA, so that is an innovative new drug, is when the FDA grants that approval for a company or sponsor or a program lead to start testing that particular therapy in humans. So phase 1 is generally for safety first. So the first is really in much larger therapeutic areas. The drug is tested in healthy adults to begin with, and this is usually just to see safety and tolerability and how that drug responds. So we talk about PK, and so pharmacodynamics and pharmacokinetics. So that is, how does the body affect the drug and how does the drug affect the body? So we do this in healthy subjects in phase 1. However, in the case of rare disease, because our patient numbers, our patient populations are so small, and every child and every patient is hard to find and precious, what we do in rare disease is we combine this into a phase 1-2. So phase 2 is typically where we start to look at the efficacy of the drug. How efficacious is this drug in treating the symptoms of the disease and the root causes of the disease? In rare disease, we combine these. So generally, a phase 1-2 is a combination of looking at both the safety and tolerability of the drug and then some efficacy measures as well. And then we move into, so phase 2-3B is really what we call in the rare disease more of the pivotal study, which is the data is taken from a larger cohort of affected children or affected patients. And that is the data that's collected on the efficacy or the effectiveness of the drug, and that is what will be submitted as a package of data to the regulatory agency like the FDA in the US or the CMA in the EU. And that is determined, that data is what determines whether or not that drug will be approved for use for what we call the indication, so the use that the drug is intended for, approved. So that's the phase 2-3B. Once we get past the regulatory approval phase, and there is one therapy for Batten disease right now that is past this regulatory approval stage, and that was BioMarin's Brineura or solipinase alpha. That was approved by the FDA for the treatment of CLN2 in 2017, and then in other markets around the world following that. Even though the phase 2-3 is over, we call this a phase 4, and this is the real world now. This is where we look at what does an approved drug look like in the real world outside of a clinical trial setting. A phase 4 study, really looks at continuing to collect that data in patients in the real world setting. And so we'll talk a little bit about each of these phases today. Phase 1-2, phase 2B, or into that phase 4. And each of the companies that are speaking to you today, or program leads that are speaking to you, will talk about which phase their particular program is in. Very quickly, the drug discovery process can be long and can be very expensive. And so the external pressures on the research and development of these molecules to get them from the lab into your children is really an expensive and long drawn out process. And as we sort of touched on at the end of the last session, is these pressures are really sometimes well beyond our control. It's subject to FDA or the regulatory bodies. It's subject to the economic situation. We know the biotech market in particular has suffered proportionally more than other sectors because of the global financial crisis, and then within that the rare disease sector suffered even more. So a lot of the companies here today could speak through clenched teeth about how difficult that process can be when you're at the whim of FDA and economy and other pressures. But essentially for every 10,000 molecules that are looked at under the microscope or through the laboratory lab bench, only one of those 10,000 on average will make it all the way through to regulatory approval, and that process can cost up to $2 billion to go from that very early discovery phase all the way through to regulatory approval. So it's an expensive process, and certainly that clinical development phase there, that stage 3, is for the most part the most expensive phase of that pipeline development as well. So it's really just to give you some context around the development process, and now we're going to jump into the clinical trials and the programs that are currently active. A couple of definitions that you'll hear. So generally a clinical trial is something that is initiated and sponsored and led by an industry partner or a company, whereas an investigator-initiated trial is an independent investigator that might use the sponsor's drug or use the sponsor's intervention, but it's really at the discretion of and the instigation of the researcher or the clinician to begin with. So we actually have a combination of both clinical trials and IITs that we'll be talking about today. So without further ado, I will hand over to Dr. Liz Berry-Kravis, who joins us online from the Niemann-Pick C. She's in hot demand. I think our first thing to try and do with the next grant is to clone Liz and have her presenting all over the world. Yes, we could do with more Liz Berry-Kravises. But Liz, if you can hear us, I'm going to hand over to you now.
Okay, can you hear me?
Yes, we can.
Okay, all right, great. So I'm going to do a whirlwind trip through gene therapy for CLN1, and I'm just going to go over an update from our program. You're advancing the slides. Okay, can you advance the slides? Okay, so as everyone knows, we got permission from the FDA to do an expanded access or compassionate use. This isn't a trial. It's viewed as compassionate use to treat one patient with CLN1 with the TASIA gene therapy for CLN1, TSHA 118, which the program had been on hold because of financial reasons and all the biotech crunch. So we dosed that first patient in October of 2023. Since that time, we've done a few things. We submitted all our safety data to the FDA and asked them if we could treat more patients by changing our expanded access program to an intermediate sized patient population expanded access. And while we were working with them on that, our patient lost vision, and so we got an emergency IND for retinal treatment of the initial first patient, which we did. And then the FDA did give us the go-ahead in August of 2024 to do an intermediate-sized patient population expanded access and treat more patients. Then it took a long time to get through our IRB and our contracts people, and ultimately we did that this spring and then treated a second patient. And right now we have 3 patients in screening that are scheduled to be dosed in August. Okay, next slide. These patients are very early in disease and they're the juvenile onset form, mainly because the gene therapy only works well if you treat very early in disease. This is just our protocol, and I'm just going to point out that we try to get testing before we actually treat the patients to get an idea what their trajectory is. We do the gene transfer, and then we do a lot of tests depending on what the patient can do to try to monitor how they're doing clinically. We do MRIs to monitor safety, LPs periodically for CSF analysis to make sure we're not having inflammation, and to measure PPT1 and CSF, which Steve Gray has collaborated with us on. We do the standard Batten scales, the UBDRS and the late infantile Hamburg scale, and safety labs for antibodies, viral shedding, and then PPT1 also in blood. Okay, next slide. So our first patient did pretty well after the infusion. He has a little kind of meningitic symptoms, which is common after gene therapy. He had some side effects of steroids, which are expected. He seemed to stabilize for a little while, but then after a few months, he lost vision very rapidly, which made it difficult for us to kind of assess him. He had these falling spells that don't have an EEG correlate. And he's had some cognitive and language progression for sure, but some things are hard to sort out because of the vision loss. And these are just his UPDRS scores shown to be going up, particularly in the physical area, but some of this is because he can't do some things because of loss of vision that he could do before, and he's not always cooperative. We did and his Hamburg scale has even come down just a little bit, so a little bit of progression. We did the emergency IND to treat the eye based on the CLN5 sheep studies for intravitreal treatment. It took the FDA a long time to hem and haw over that, but eventually they let us go ahead. So we used a systemic steroid boost and continued sirolimus, used ocular steroids, treated him in the eye. Unfortunately, the vision was sufficiently far gone that we didn't really see an improvement. But on the positive side, there was no immune reaction, no systemic reactions, and just a transient increased ocular pressure from steroids. So that went pretty well as a second AAV9 exposure. Okay, next slide. We just treated the second patient, and his course since the infusion, which is only really about a month ago, is that he had a few little things right after the infusion, and again, the meningitic kind of symptoms, but he also got stomach flu, which complicated the whole thing. So we had a lot of vomiting and related symptoms in the first week that we treated, and then he recovered from all of that. He had onset of seizures after we transferred him back to UT Southwestern, which is they actually got our protocol through their IRB, and they're gonna do all the follow-up there, so we're collaborating very nicely together. And then he's had steroid side effects, and we'll be coming off steroids soon. And just as of yesterday, a new update from Steve Gray's lab, they've analyzed the CLN1 activity in spinal fluid that you can see on the chart here, and you can see both of these patients had very low activity before they were treated with the gene therapy. The controls, we have other disease controls that don't have CLN1 in the gray box, and that shows your range there. And then both patients have normalized levels of CLN1 activity post-treatment. Next slide. And so our conclusions so far are that the 1E15 vector genomes dose that we gave appears safe based on N of 2 so far. It's hard to know the precise efficacy of the first patient due to the vision loss, but there may be slowing of progression, maybe not. We need longer to see what the stability is long-term. The PPT1 levels are normalized in CSF and blood. Did we treat too late for patient 1 to stop the progression? Do we need patients that are pre-symptomatic? Those are questions we need to answer. When we give PPT1 through an LP, where is the enzyme we're seeing being made? Could it be being made mostly in spinal cord and brainstem? Should we use a different delivery method? These are questions we need to answer. The eye was too far progressed to show benefit from TSHA 118, but we set 2 precedents, which are that it appears to be safe to give the gene therapy to the eye after already giving it in the CSF. There was no major immune issue. And we can use the same construct, except with a different gene, as a model to dose a gene therapy. So we're working on getting IVT dosing guidance for CLN1 now from the sheep model. We'll need to explore all these questions going forward, and we will be doing that with the upcoming patients. Next slide, and I just want to acknowledge Teja and Steve Gray and Sharon King for funding the early work, as well as our coordinators at Rush and all of the other services we have to work with at Rush, like pharmacy, anesthesia, and ophthalmology, and the patients and families, and the FDA, which was actually pretty collaborative for all of this. Okay, that's it.
Thank you, Liz. Great update, and I know some of that data was hot off the press late last night, so thank you so much. Will you be joining us online as well for the Q&A in about 45 minutes?
45 minutes. Yeah, I should be able to do that.
If you're still around. We know you're in the middle of a conference there too, so we hope to see you, but no problem if not. We have Marissa as well, I think, on the line that can help. Okay, so I am... Oh, can I have the control back, please? There we go. Okay, so we have a large BioMarin team in the house who will be joining us on stage for Q&A, but I'm going to speak a little bit not only to the sponsored programs, but some of those IITs that I talked about as well, so I'm going a little bit off-piste to what the company would necessarily talk about, but I'm going to give you a quick overview. In terms of the Brineura, as most of you are more than aware, this is the one approved therapy for any type of Batten disease. This is the intracerebroventricular infusion that happens every 2 weeks for children with and adults with CLN2. So this was, again, as I mentioned, approved in 2017. We have, in the meantime, ongoing studies. So the phase 1/2 that was completed not so long ago was actually the extension study to evaluate the long-term and safety. This was published by Dr. Angela Schultz and others from the leading site, so Dr. Emily Delis Reyes at the Nationwide in Ohio, the Gosh Great Ormond Street Hospital in London, and the Italy site, the Bambino Hospital in Italy. So these were the 4 leading sites that have continued on the research with the observational studies of Brineura. This 5-year extension data basically showed that the safety and tolerability of Brineura over a longer period was retained. That safety and tolerability continued, as did the efficacy. So we saw this continuation of the stabilizing of the motor and language score, particularly for those that had started the therapy at the younger stages. And of course, the sub-analysis from that shows that any children that started pre-clinically, so pre-symptomatically, so because they had older siblings that were diagnosed, they were able to be diagnosed before the onset of symptoms, are doing particularly well, and it looks like it's delaying the onset of the condition, of the disease symptoms altogether at this point in time. Of course, this ongoing real-world data collection that I talked about as well is this sort of phase 4 of the study. Groups like Emily Delis Reyes and Angela Schultz are collecting this ongoing work, and the data, for example, presented at World only in February this year by Angela Schultz's group, showing that in the real-world setting, Brineura seems to be keeping the seizures well under control, and certainly the movement disorders. So they examined myoclonus and dystonia, and although the dystonia tended to still come on despite treatment, the myoclonus is seems to have an improvement in that over the course of the treatment so far. And then there's the ongoing phase 4, which is this 10-year prospective observational study. So really important to continue. You know, once a drug is approved, that doesn't mean we've learned all we need to know. It's really just the beginning, and we really, you know, we're in an uncharted territory with this approved therapy, and we want to keep learning about this drug, learning about its treatment effects, and learning about, you know, the children who continue, and adults that continue to receive Brineura. So in terms of the ocular ERT, again, Dr. Emily is in the house at the Nationwide Children's Hospital, are currently in the midst of an ocular, so this is where injections are received intravitreal, so into sort of the fluid or the jelly-like substance into the eye every 4 weeks. This is fully enrolled. This was by invitation only, this particular study, and it's randomized, so one eye is injected and the data is collected over this time. Efficacy data hasn't yet. We've seen some safety data to show that it's safe and well tolerated, but we haven't yet seen the efficacy data from this study because we're still collecting that. But Emily's team will be presenting that. Dr. David Rogers is the ophthalmologist from Nationwide Children's, will be presenting that later on this year, so we look forward to seeing that. Heading a little off-piste with the IITs. Why is this not advancing? It doesn't seem to be advancing. Oh, amazingly, there's no trapdoor. Pause the clock. That's weird. It was working. It still says... Now the laptop's frozen. Oh, no. There is no rhyme or reason. Yeah, I'm nearly done. Oh, it's done it again. It's stuck on the same. Let's see if I can do it this way. That is so bizarre. I know, I've jinxed us. Let me try. Let's try this. Hopefully I don't have to do that for every slide. But this is something that you would all be familiar with as well. I've presented this last year. That this is the ocular ERT that is the investigator-initiated or investigator-led study happening in Gosh, Great Ormond Street in London. This is a study that has since been published, and effectively from there, so this is really what we would call off-label use of Brineura. The intravitreal treatments have also been done, and a case report in Germany from a single child, and it's something that we're receiving a lot of questions about as a Centre of Excellence consortium from families wanting to explore this for their child as well. So we will talk more about that. We have an Ask a Doc tomorrow, so Dr. Emily Delastreyes will be on that panel. She'll be up here for the Q&A at the end of this session as well, as will the Biomarin team. We have representative from the company, so feel free to ask questions about that. I really just wanted to present this to you as an overview of where we're at with Brineura right now. Over to our next speaker, Suze.
Hi everyone, my name is Suzanne Plaisir. I am the head of clinical operations at Lattice Bio, meaning I'm responsible for the preparation and execution of the clinical trials. I want to thank the BDSRA Foundation for having me here. It is a real honor. And I have to say I've presented a lot in the past, but never for an audience that mattered as much as you guys do, so I'm not nervous for sure. I will be presenting the clinical gene therapy update for LTS 101. It's in development for CLN2, right here. I'm an employee of Lattice Bio. Lattice Bio is a company located in both Philadelphia and Boston and working on a one-and-done gene therapy for rare neurological disorders. We are, sorry, I skipped over the first bullet here. That's my nerves getting in there. We're founded by Bev Davidson, who heads up a research lab in Children's Hospital Philadelphia, CHOP, and she's a world-known leader in gene therapy. As mentioned, we're in Boston and Philadelphia, working on that one-and-done gene therapy. And we have a platform with capsids, adeno-associated virus vectors, that are modified to deliver genes into the brain. So technically, we use a virus vector as a vehicle to deliver specific pieces of DNA in specific regions of the brain. So technically, we have a different vector for the different brain regions delivery. Our pipeline currently is developing programs for CLN2, that's why I'm here, and Huntington disease. For all this research and development, we are supported by exceptional teams. Already mentioned Bev Davidson and her research team at CHOP. We work together with Dr. Angela Schulz from the UKE in Hamburg, Germany, and Dr. Backe Ernst-Nicholas from CHOP. They're sitting on our scientific advisory board. And also, we are very appreciative of the warm welcome and partnering with the BDSRA Foundation and the engagement of the Centers of Excellence already mentioned earlier. Diving into one slide on the clinical update. So for our program, CLN2 program, we have engaged with both the FDA and the German health authorities, known as the PI, where we discussed our CLN2 program, and both organizations have provided supportive feedback. We are currently working on preclinical studies to enable IND submission. I think that was explained a little bit before, the IND submission. And our next step is to navigate the regulatory landscape to get submissions and approvals done for this study in order to start the clinical trial. I want to make sure that I address that we currently have in mind what our project and our protocol should look like, including the patient population, the eligibility criteria, and also ages and other matters. This is our proposal. This will have to be discussed with the agencies, and we have to get approval. So although maybe some statements have been made, I think we cannot really disclose or say what our patient population will be unless we have this approval. We will, as soon as we are ready to speak about this, engage through different ways of resources to make sure we reach you with the updates for this study. And as you might know, it will take a village to get these developments and research done. So pictures of our team both in Philadelphia and in Boston, and also reflecting on our collaborations here. Thank you for your time.
I hope it works for me. Oh no, I jinxed myself. I'll just do it from here. Okay, hi, I'm Chris Onsman from Turn Therapeutics. I'm joined by Jessica McLeod, our Director of Clinical Operations. We're very proud to be here today. Sorry. Turn was launched 10 months ago, so we're a young company, but we're not new at CLN2. So my partner and co-founder, Alex Bailey, who is our CEO, and I have been developing these particular CLN2 treatments for quite some time, 6 years for me and longer for Alex. We were previously at Regenix Bio, and we in-licensed these programs when we founded Turn. So our sole reason for existing as a company is because of you and for you. And I'm going the wrong way. Okay, so we have one single gene therapy that is formulated to treat two different parts of the body, the eye and the brain. But it works the same way in both areas. So just as you've heard from other people developing one-time gene therapies like ours, we use an inactivated virus to be sort of like the FedEx truck that carries the gene, the CLN2 gene, to the affected cells. So in this case, the retinal cells and the brain cells. They're the cells that take up the virus, produce TPP1, which is what they were unable to produce before because of the CLN2 disease. But in CLN2, we're lucky that TPP1 is also secreted so that the neighboring cells can use it as well. And so we were able to treat the entire retina through treating just a small part. The secreted TPP1 reaches the rest of the retina, and the same thing, we reach the entire brain. Our pipeline is shown here for our 381 program, which is our ocular program. We are far along in our phase 1/2 testing. We have completed our first 2 dose cohorts, and we are well into enrolling the expansion cohort. We're calling this our phase 1/2 study. We don't speak publicly about what our regulatory plans are, but please rest assured that we are using every regulatory shortcut that's available to us legally. And we recently did receive RMAT designation, and that gives us additional opportunities to speak with FDA to align on the path towards approval. Our 181 program is not as far along. However, we have treated a single patient, and I'm going to show you a little bit of data from both. But because I'm limited to 4 minutes for 2 programs, I can only give you a snippet. So please use the Q&A to ask me questions so that I can tell you more. So for 381, the most important thing, first of all, is safety. We have a great safety profile. Okay, all right. So safety was good. We definitely saw TPP1 expression in the eyes. That resulted in photoreceptor, which are the rods and cones, the affected cells in the retina, preservation, and I'm going to show you graphs of that in a second, that helped the children to then function visually in the world. So here, this is maybe not as fun of a way as I could have shown you with more time, but this is not showing up correctly on here. But the blue lines are the untreated eyes, and you can see that the degeneration is continuing. And the orange lines are the treated eyes, and you can see that in all, well, you can mostly see that in all of the cases, we are either holding the retina steady or even seeing some improvements, and that the improvements were unexpected. We're very encouraged by that. Our 181 study, as I said, this is a single patient so far, and similar safety, increased TPP1 that looked just like the graph that you saw earlier, where it peaked and then stayed steady for 2 years in this child. His seizures were reduced by 90%. Through 2 years, he was able to stop taking 2 of his 4 seizure medicines and to stop taking Brineura. And we saw some stabilization or small improvements in fine motor and language skills at 2 years. And this, you can see the before and after of his seizure activity, so you can see how well he responded in that way. And so I just want to leave you with the message that we are here for you. We are moving as quickly as we can. We share updates through BDSRA. There's a lot more going on behind the scenes than we can always share publicly, but rest assured that we are working hard and urgently for you. Thank you.
All right. Let me see if I can figure this out. Yes. Also doesn't look exactly the same, but it should be okay. I don't have a lot of graphs. Again, my name's Kat Meyer. I'm the CSO of Alcyon Therapeutics. You probably haven't heard of us. It's a small company that I recently joined. We have our own therapeutic pipeline, including one-and-done gene therapies, as well as small molecules that are preclinical stage that target neurodegeneration in a broad manner. In addition to that, I'm not going to talk too much about it, but one thing that really drew me to this company is their interest in improving the delivery of the drugs to the brain. So we have an entire team that works on hardware, catheter port systems, shunts, etc., but also studying the CSF dynamics and how we can use those CSF dynamics to get the drugs to where they need to go. And obviously, we're planning to leverage that also for Batten disease. So what we developed back in the day when I was at Nationwide Children's Hospital, in collaboration with Sanford Research Jill Weimer's lab, is a CLN3 gene therapy It's again one of those AAV viruses. The virus that we're using is quite de-risked. It's been used in the clinic for a long time for Zolgensma, for example, which is another approved therapy that has been used in thousands of patients. It will allow to make the protein inside the cells, because again, for CLN3, you probably know that that is important. It's a lumbar intrathecal injection, so we don't make holes in the brain, etc. It's directly into the CSF and spreads from there to the brain. This program was previously licensed to Amicus Therapeutics, and we are very grateful that we are able to take this over now. So you've probably seen some of it already, but I just want to kind of reiterate the clinical trial. We have treated 4 patients so far. There are 2 dose levels. Dose level 1 was 6013 viral genomes per patient. Dose level 2 is 1.2E14 viral genomes per patient. So this is important per patient, not per kilogram, because some of the toxicity that has been seen in the clinic in other trials is dosing at a much higher level intravenously, measuring the doses per kilogram body weight. Below you can see the patients that we've treated in the trial were all symptomatic. They were 3 to 5 years post-symptom onset. And just a little illustration on where we are now today. So the star indicates the time of treatment, and then there should be a gray box here that now shows you the age of the patients currently. And I'll be able to give you just a few updates here. We don't have a lot of time, as you know. We've seen encouraging results in all 4 patients. So first of all, safety has been outstanding. We haven't seen any issues, including liver enzymes, et cetera, as well as the 3-year follow-up on the UBDRS, which is the length of the clinical trial, really showed a very strong sign of potential efficacy, I would say, in the patients that we looked at. And then this, I'm sorry, this wasn't like that when I made the slides, but okay, so there's additional data that we've collected. As you've seen, the clinical trial was started in 2018, so we have some data from some of the patients that are up to 5 years post-treatment now. And 2 of the 4 patients that were treated still continue to show stability on the UBDRS, so they haven't progressed any further since the time of treatment. And also 2 of the 4 patients are seizure-free at the moment with only 1 seizure medication. In addition to that, we have some limited cognitive data as well from the 2 patients. And it really indicates that the patients are continuing to learn and have an ability to remember items in a story. And patient number 4 that received the higher dose has cognitive abilities that are in the normative range, which we think is really encouraging. So what are the next steps? Again, this isn't me. This had all the same font when I put it on there, but it is important that we are manufacturing an additional clinical vector lot. So we are, in order to continue the trial, we have to make more material. And that's what's happening right now. And if everything goes well with the manufacturing, we should be able to enroll additional patients in the trial in Q1, Q2 2026. We are seeking regulatory input for, again, as previously mentioned by my colleague, it's very important actually to get, we can design our clinical trials the way we want, but we do need to get agreement and alignment with the regulatory bodies in order to make sure that what we want to do is actually getting us to approval. And that's, again, why the natural history studies are so important, because we need to be able to show to the FDA and EMA that the treatment is working. Is very hard to do if we don't have the natural history data from patients that weren't treated. There are some backwinds to this program that weren't there a couple years before that make us really confident that we can move this fast. Number 1, we are really grateful that Amicus continues to support this program. They are sharing their developed reagents, raw materials that they made. They give regulatory feedback that they have received to date. So this is not something that they have to do, but they are willing to do to continue to support the Batten disease community. In addition to that, the AAV gene therapy field has undergone a lot of maturation. I would say maybe we're not completely mature yet, but a lot of maturation when it comes to the manufacturing. And we can actually now use the same manufacturing platform that was used for the first 4 patients. And that gives us additional backwind in order to use that data that we've already collected with the FDA. And then, as I mentioned, continued development of natural history and potential biomarkers that we can use to support the results that we find in the clinic. So, thank you very much.
It's me again. I'm sorry if anyone's getting sick of hearing my voice, because I sure am, but sorry, we have another presentation from me. Mary Beth Kaiser is our President and CEO of the Beyond Batten Disease Foundation. Her name is in the program, but I'm also, as full disclosure, I'm a consulting scientific officer to the Beyond Batten Disease Foundation as well. So she's asked me to speak to the data around this program. Mary Beth will be up here with us on stage for the Q&A as well. So in terms of the miglustat treatment, so now we're working our way through the different subtypes from CLN 1, 2, 3, if you haven't worked that out in the program. So in terms of the CLN 3 disease and the miglustat treatment studies that are currently ongoing, we have presented this data again last year. This is from the phase 1/2. So miglustat is effectively a repurposed drug. It's currently approved for other similar lysosomal storage conditions, namely NPC and Gaucher's disease. This is a drug that is, the mechanism of action is to clear, you would have heard about these GPDs, which are these, glycerophosphodiester fatty accumulations within lysosomes of cells in these lysosomal storage conditions, including CLN3. And we believe that the GPDs are being reduced by the miglustat. And Dung Do, who you saw earlier from her presentation, shows that these are reduced with miglustat treatment. So we believe this is the mechanism of action. You also heard about the neurofilament light chain from some of our earlier presenters as well. So from the phase 1/2 safety, which continued on from what we'd seen in the mouse models of these of CLN3 disease, our phase 1/2 safety, which was carried out in 7 adults with CLN3 disease, showed that there was a considerable reduction in neurofilament light chain. And as well as the GB3, which is a type, it's a sort of a subtype of these GPDs. So those kind of key markers were shown to be reduced in the treated patients over the 12 months, which was again seen to maintain that decrease over 18 months of treatment. And in addition, at the 18-month time point, we saw that a progression of motor symptoms had slowed considerably, as evaluated by the modified UBDRS, which is the clinical rating scale that is typically used for CLN3. At 18 months, the change from baseline, so for had only increased by 1 point. So the higher the score, the greater the motor impairment. Where it had increased by 1.83 points over those 18 months. In untreated natural history controls from the Rochester University Batten group show that typically over the same course in the same age-matched controls, you'd be looking at more like a 6-point increase in score over that time. So it seems to be a stabilization. These are very small numbers. This is only 6 patients, so it's not powered to statistically prove any efficacy, but it was just an interesting signal, both the neurofilament light chain, the GB3 deposits, and then also in that motor score. So that data collection is now complete, and we are in the process of putting that manuscript together. And those 7 patients are actually still receiving that on compassionate grounds into the foreseeable future. The big question is the phase 3 pivotal. So in these pediatric study now, we're pending commencement of that. We have had unanticipated delays due to funding, nothing to do with the clinical data. It's just been a real push. We've talked a little bit about the FDA requirements for placebo-controlled arm. that's been the challenge with this particular program, is that they had initially mandated a one-to-one, so one treated child to one control placebo-treated child. And that was very hard. That basically doubles the size of the trial that you need. Made it very difficult for us. But what we have found over time in the last year or two is that there's been an increase in off-label use of miglustat, meaning families are applying to have miglustat treatment for their CLN3 children. And we have been very rigorously as possible collecting the data from baseline and then intermittent assessments on visual acuity and UBDRS scores, neuropsychological scores, to see what the effects are of miglustat on these children. And what we found is actually quite an interesting effect. The FDA had asked for a primary endpoint of visual acuity, and as it turns out, it looks like in the younger children that visual acuity seems to be either preserved, or in some cases, if treated early enough, there seems to be an improvement in visual acuity. So this has actually enabled us to pivot somewhat in our discussions with the FDA. The most recent discussion, was the June 2025, just last month, to talk about the need for reducing that placebo arm. We think, I don't know if we can completely eradicate the need for a placebo arm. They may still ask for some placebo-controlled on there, but it will definitely reduce the size of the trial and therefore the cost. And we have, I guess, encouraging results that suggest that we could meet this primary endpoint, and particularly, in a smaller time frame as well, if we run the trial well designed and with the right recruited patients. So that's a brief update on the miglustat, and Mary Beth and I are happy to take any questions in the meantime in the Q&A. I'll hand over to Dr. Shiloh Malolsky.
Hi, I'm Yael Shiloh-Malowski. I'm a neurologist at the University of North Carolina at Chapel Hill. I'm also the director of the BDSRA Batten Center that we were designated a year ago, and I'll present the Zebronkisen, which is a personalized antisense oligonucleotide for a very rare variant of CLN3 Batten disease. It was named Zebronkisen in honor of the zebra and monkey that comforted Mackenzie and Amelia during the doctor appointments and snuggled them every night. This is the a partial list of collaborators and people working on this study. So it starts with the For Batten Foundation, that is sponsored research in this field, and then put together the N of 2 project, which is the treatment of 2 patients with this rare disorder. And I'll mention Tiffany, who's here, and will speak next about the next project, but also Michelle Hastings, who's the scientist that did all the work that led to development of this ASO, and she's with University of Michigan. So the Zebronkisen N of 2 trial enrolled 2 girls, twins with this rare, ultra rare variant in CLN3. They were 14 at the time of enrollment. They were dosed with the drug. It's dosed through lumbar puncture every 3 months with dose escalation, gradual dose escalation through the dosing period. The study looks at safety monitoring and efficacy, and you can see some of the measurements that we are following, including global improvement, 2-minute walk, extensive physical therapy, gross motor abilities, the Unified Batten Disease Rating Scale, Violent, which is cognitive adaptive behavior, quality of life, and behavior checklist. We also follow biomarkers. And we're now at the 1-year
time. The first dose was in June 2024, and then the 5th dose at 1 year was just last month. And we're able now to report 5 doses of safety and 4 doses of efficacy measures. So as far as safety, adverse events, there were some transient insomnia and agitation following the drug dosing, but no severe adverse events related to the study drug and no lab abnormalities. As far as outcome measures, the global impression showed improvement in both girls, and the 2-minute walk, which just one of the girls is ambulating, and she was able to increase from 22 meters or yards to, for the American, from 22 meters before the start of the study. After 6 months, she walked 48 meters in a 2-minute. We also saw stability of the violent, the cognitive abilities, and stability or improvement in the behavior and quality of life. So to sum the first year of this drug, we're seeing that both girls are experienced no loss of abilities and showed some gains. It was well tolerated, and we saw only transient and no severe drug-related adverse events. So these photos are all part of videos, so it's images from videos, and you can see on the horse on the left side, Mackenzie had a lot of dystonia, a lot of days of dystonia, not every day, but many days where she would be leaning forward. And following the drug dosing, she had for times she would have dystonia completely disappear after the drug increase, and you can see how the instructor is able to walk away from the horse, not even holding her, which was new, was not possible before this drug. And then you can see Amelia. These are the image kind of to the center. She's eating, I think it's a candy, and she's smiling. She's happier. She's clearly happier since she started being on this drug. And on the far right, you can see her putting a straw in her mouth and taking a sip. She did not take food or drink for about 2 years before starting this drug. So to sum the first year, going into this trial, our hypothesis was that the ASO, while through restoring the CLN3 protein function, would slow down or stop progression of loss of function that is typical for CLN3 Batten disease. But what we have seen in the first year was not only stability, but also unexpected improvement in several domains. So we're encouraged with these results, and we're currently working on expanding this approach of ASO in treatment of other CLN3 patients.
Okay. Hi, everyone. My name's Tiffany Sepp. I'm the CEO of Vanguard Clinical. We're a contract research organization in San Diego focused on rare disease clinical trials. On a personal note, I'm mom to these 4 kids here in this picture and these giant dogs. So I had the privilege, as Yael mentioned, of being the development team leader for Project Butterfly, the NF2 ASO program that you just heard about. That project just completely changed my life and my career, and so it definitely sparked for me a need to do more and brought me to my next chapter. So in March of this year, I launched Vanguard Clinical Rare Disease Foundation, which came from everything that I learned on Project Butterfly. And the mission of VCRDF is to summarize all these words on the right, to just reimagine radically the rare disease drug development process and make it faster and more efficient and ultimately scalable so that we can deliver treatments to your kids. So taking the roadmap that we created for Amelia and Mackenzie, it's our hope that with each new ASO that comes through VCRDF, we can smooth out the bumps in the road and make the mountain a little bit easier to climb each time. So the first project that we're taking on through VCRDF is an ASO for the common CLN3 mutation, and we're developing this by building directly on the playbook that we established with Project Butterfly. The ASO was also developed by Dr. Michelle Hastings at University of Michigan, and the initial science was funded by the Batten Foundation that sponsored Project Butterfly. And so Batten Foundation has now passed the baton to VCRDF, and our goal is to follow that same little bit of a shortcut with the FDA through the N of 1 guidance document. And we are going to start with 2 children, a pair of siblings, also Yale's patients, because we've already gone through the UNC process and we know what that looks like, and follow the same FDA pathway with the N of 1 guidance. So we're starting with these 2 boys because it will be the fastest and the cheapest, although still incredibly expensive, way to get the drug into the clinic. And then our plan is to start with the data for those 2 boys, get the initial dose in, and then submit an amendment to the FDA to hopefully expand our clinical trial to treat more kids with CLN3. We did have initial engagement with the FDA in a pre-IND meeting, and they confirmed alignment with that plan. So we are optimistic that once we have the funds, we can quickly execute on that strategy like we did with Zabrunkicin. We did launch our fundraising campaign and submitted an application to the NIH for an urgent grant, which is not very urgent, by the way. And we know that we can do this efficiently, safely, and effectively as soon as we have the funding in hand. So I think I was under 4 minutes. Thank you so much, everybody, for the opportunity to be here.
So unfortunately, Neurogene weren't able to be with us this year, and so I am presenting slides on behalf of the company. I'm not a consultant for any of these other companies, I should add. It's only beyond Batten disease. But in any case, this is a study that I think has had a really sad development recently. It was, for all intents and purposes, the leading gene therapy trial for the Batten diseases in all the different subtypes. It had really, despite the biotech downturn in the last 2 or 3 years, this had continued reaching its milestones in terms of getting the IND, starting recruitment for the trial, moving through the different phases or the different stages of recruitment. And unfortunately, in November, it had a little bit of a hurdle. That's not the end of the program, but I will talk to that in just a second. But a little bit of background. This is a dual route of administration. It's the only kind of its kind in the gene therapy Batten disease landscape, meaning that there is 2 routes. So they have both an intravitreal and an ocular IVT and ICV dual route of administration that is given in the same procedure, well, sequential procedures, but the child receives both routes of administration, whereas our other programs like TURN, for example, has one program for the ICV brain infusion delivery and another program for the eye. The sites that are recruiting or were recruiting and involved in this study and continue to monitor the patients are in University of Rochester and the Great Ormond Street in London. These were 6 patients recruited from the ages of 3 to 9 years. It is an open label, so there's no placebo control for this study. The first 2 children were treated in this low-dose cohort, which was then escalated to a mid-dose, and the final 3 children were treated at that high dose. They had a meeting at the end of last year to discuss the requirements and were fully intending to move to this phase 3 pivotal clinical trial. The data was good. The preclinical data from the sheep models that are in New Zealand are best in class in terms of large animal models. These sheep are doing extraordinarily well. those high-dose sheep that are now, 3 or 4 years post-treatment. In terms of sheep age, it's really good. Unfortunately, in November, Neurogene announced that they were not expecting to move forward with the program. This is really personally very hard, to hear this news when you have so much, and I know the kids in this trial and the kids are doing well, that given the rarity of the disease, unfortunately, Neurogene had predicated the development of this program on getting what we call this streamlined registrational pathway, which is called the RMAT, or the Regenerative Medicine Advanced Therapy application. That TERN, so Dr. Chris Onsman just mentioned that TERN have been given the RMAT designation for 381, which is actually a really big deal. Doesn't mean it's approved, but what it's given is basically a green light from the FDA to say, hey, we can see encouraging results from this program, and we want to give you a smooth path to get in touch with us and keep in communication with us so that we can help you get this to the finish line as quickly as possible. Unfortunately, this was denied, although Neurogene believed that they had met the standard of preliminary clinical evidence required. And at the moment, Neurogene is reconsidering, how to get this program forward, because it does matter to them. They've stayed in touch with us since this news and all through the development of the program, and had asked BDSRA to be involved in some pivotal meetings to try and see what way could we move forward with this. And that is still under consideration at the moment. So unless someone has a big bag of money, and this is the problem, trying to get these programs moving forward in the rare disease space is really hard, even with the best intentions from the company. So that's an update for Neurogene, and they have their patient, they did give me their patient advocacy email address. If you have any questions and you're a CLN5 family that want to hear more, please reach out to me or to the company directly and find out from them. Back to you, Tiff. We just need like a little baton.
Hello again. Okay, so in addition to my work on CLN3, I have recently joined the Charlotte and Gwyneth Gray Foundation to serve as their development team leader for the relaunch of their CLN6 clinical trial, and I'll talk more about that in just a few minutes. Well, in a minute. Kristen couldn't be here today, but she will be on Zoom for our Q&A, and so I'm here on her behalf to share the foundation updates. So just a little bit about the foundation first. So since its founding in 2015, they have been at the forefront of pushing research forward for CLN6. They funded and launched the first CLN6 gene therapy trial in 2016, which treated 15 children. And in addition to the gene therapy program, the foundation has supported small molecule discovery with Rarebase, early stage work at TIGEM in Italy, and has built meaningful collaborations with many leaders in the field. And they're not stopping there with just one approach. The focus right now is to accelerate the next clinical trial for CLN6, but also they're making investments in research to improve gene therapy itself, looking at new dosing strategies, delivery routes, and ways to make this more effective overall. Okay, so 2025 has already been a really big year for the foundation. In January, we successfully regained licensing rights to the CLN6 gene therapy program, which gives the foundation control to move the program forward with urgency and care that you guys deserve. So when the original trial was conducted, it was proven safe, but did not achieve the level of efficacy that was hoped for. So that trial gave us some essential data and insights and lessons that we've used to inform the path forward for the new trial. So our foundation is now collaborating with a powerhouse science team, Dr. Jill Weimer and her lab, Dr. Ray Wong at Chalk in California, and to develop a higher dose treatment strategy designed to improve outcomes with children with CLN6. We are also, the Gray family made the decision to donate Charlotte's tissue samples to Tim Yu's lab to help to advance the science and to better understand the results from the nationwide trial. In June, so last month, just a couple weeks ago, we submitted a major grant to the California Institute for Regenerative Medicine to help to support the next phase of the trial. And we've signed a contract with our vector manufacturer to begin producing the new drug. And our goal is to relaunch the trial in early 2026. Last July, Charlotte Gray gained her angel wings, and her life and her light continue to guide everything that we do at the foundation. Kristen and Gordon Gray continue to lead the fight with courage and conviction. Kristen is one of the strongest women I think I've ever met, not just in memory of Charlotte, but on behalf of every single child still waiting, and yesterday was Gwen's birthday. So to wrap up, we believe that families are not bystanders in this fight. We believe that every child's story and every family's voice and every effort matters, and together, not just hoping for a cure, but building one. Thank you.
And finally, on to CLN 7. So this is program slides provided to me by Elpida Therapeutics. They were supposed to be here, but unfortunately, due to actually an urgent FDA meeting, which is a positive thing for this program, they chose to attend that, and I'm presenting on their behalf. But it's a positive news story. Bench to Bedside. This was a drug that was developed at the UT Southwestern under the auspices of Dr. Stephen Gray, a name familiar to many of us, and Dr. Jin Chen, led by the clinical neurologist Dr. Burj Manassian. So this was a study that was first done in the CLN7 mouse. It was shown that this AAV9, which replaces the protein and the gene in CLN7, is known as the MFSD8. And this was shown in the mouse to improve lysosomal function, increase the protein expression, ameliorate, so reduce some of the key indicators of lysosomal deposits within those cells, increased the lifespan of the mouse, and even restored some of the behaviours. So looking at some of these behavioural mechanisms within the mouse were restored. And of course, it seemed to be safe and well tolerated in the animals too. So this is just a repeat, really, just to show the cycle to leading into the human studies, was showing first in the preclinical setting in the mouse model results, and then moving into the clinic now. So this is the dose was the 1 to the E15 per patient. This is actually one of the highest doses of AAV9 used in humans to date. And it's an IT route, meaning it's in through the lumbar, intrathecally through the lumbar, which is obviously connected to the brain. And so this was the route of administration for this particular gene therapy. They have since had their pre-IND since 2018, and the toxicology and drug manufacturing was approved. Based on that, the IND was approved by the FDA. And it has been an investigator-sponsored, so this was UT Southwestern. This wasn't a company-sponsored trial. This was an IIT carried out at UT Southwestern in 4 patients, and the data was positive. It was safe and well tolerated, and was encouraging enough that they've decided to move forward now with a phase 2B. This is just the makeup of the study. So they had a low dose patient, and then the other 3 were high dose. The data collected on these over the last few years were sufficient to move into the next phase. So UT Southwestern partnered with Elpida Therapeutics. Elpida Therapeutics is more of a social enterprise company that really moved forward. It was founded by a father of a child with another rare disease, and he really saw this as his torch to carry, and to help other families with rare diseases like his to find whatever resources he could to help programs like this that had promise to help them move forward, knowing full well, the challenges of the funding and the biotech industry. So they have moved forward. This is basically just a summary of those results. What we expect to see now from January 2026, excitingly, they're wanting to move into this full phase 2 trial now. They're planning a screening period. Sorry, the formatting of this slide also is a little funky, but they're planning a screening period of up to 4 weeks, treatment and follow-up over the course of 2 years. And this will be in around 8 to 12 subjects. Ideally, these kids will be pre-symptomatic or very early in the disease course. So the challenge here is finding those kids, and it takes us as a community to really, reach out to our global patient advocacy groups, our global family networks, even through Facebook. The power of Facebook is amazing, and these private groups within Facebook to find where those children might be around the world who are very early in the disease course or even pre-symptomatic if they've had an older sibling that's been diagnosed. And the plan is then to follow these kids over the sort of the long-term treatment, post-treatment for 3 years or beyond. And they also have a natural history study ongoing. Again, we've talked a lot about the requirement for placebo control in these sorts of studies. Ideally, there would be a natural history comparator arm so that the data that's been collected over children with the disease over the course of years is much better than having a child with, placebo treatment or a non-treatment. And generally, we try not to hold these placebo control arms in gene therapy trials. So that's why the natural history study is really, really important. So if you're a CLN7 family or no CLN7 families, please have them reach out to either BDSRA or to Suad Mesahel, who is the clinical ops manager for this program at LPDA. And yeah, we look forward to giving you some positive updates, hopefully by next year as well. And that is it from us. We are still a little behind on time, but the lunch will be provided. Don't worry, they know we're in here. They're not going to take the food away. So if you're a speaker, would like to come up on stage for the Q&A, we'll take about 20 minutes or so, or as long as it takes. We're here for you, and this is more about the content and the opportunity to ask these questions than anything. You only get this opportunity once a year, so take your time, and we look forward to your questions. I'm sure there'll be a few. Hi, Dr. Liz, we can see you online too. Thanks for joining us. And Marissa.
Okay. Hey.
And I can see Kristen is here with us now too. Hello. I'm waving to you.
Hi.
Welcome. Happy birthday to Gwen for yesterday.
Thank you. Number 12. It's pretty awesome.
Okay. John, first question.
Yeah, thank you very much. This is a question for the Alcyon people. Do you know what the inclusion criteria are going to be for your study, and are they basically the amicus inclusion?
Can you hear me?
Yeah.
It's a little bit hard to say because we're seeking right now alignment with the FDA to make sure that we can accelerate to approval as soon as we can. However, the IND is still open, and we are planning to start treating patients as soon as possible. So probably it's going to be similar for the first patients as it was. There might be some additions though.
Darn.
Sorry.
Hi, Dr. Reyes.
Hello. You're not trying to talk to me because you talk to me every 2 weeks, right? Okay.
It's the lady too, you're right. It's Christina, right? Yeah. With the vision with the CLN2 patients, have you guys done a study on the kids that have already had vision loss, and was it a progressive line, or do they just keep going down?
So the natural history is that the photoreceptors eventually in the whole retina degenerate. So there's a lot of natural history that's known. As far as whether they can be treated, our last 2 patients that I showed you the graphs of were a little more progressed than the previous 4, and we still saw good results, but they were not all the way gone. We believe that as long as there are some photoreceptors that are still alive, that we can preserve them, but we don't really know that yet. But certainly we would hope that they would respond and at least save the vision that they have left.
Okay, thank you.
Hello. So it sounds like with a lot of the gene therapy trials that are being looked at, you're needing like a population of some of the younger patients, and I know there's always the siblings that they know early, but is there any like
I guess, outreach to do any newborn screening to try to catch some of these kids for these trials ahead of time, to kind of bring a larger pool of patients?
Yeah, that's a really good question, and that is the dream, right? Is to have all of these conditions on the newborn screening. As one of our speakers earlier, I think it was Melissa, said, the requirement to have a condition on the RUSP at the moment is an approved treatment, and so CLN2 is the only one with Brineura. So really, I mean, even that we're struggling to get on the RUSP. It's not on any newborn screening programs anywhere in the world, and there's sort of units that are working around the different countries and regions to try and get that onto. But unfortunately, we're really battling those early diagnoses for that exact reason right now.
So I guess I know that I'm wondering more if like targeting specific, even just areas, to maybe even let parents like decide, do I want my child genetically screened for these rare diseases? And it wouldn't necessarily be a part of like the newborn panel, but a way to just like get these kids in. Is there any like kind of funding or like campaign to just get those kids, especially in areas where we do see some like larger populations of Batten's kids?
Yeah, it's a really good question, and I think for our gene panel this afternoon, really good kind of theoretical questions around this too. In theory, you could take your newborn baby at any time and do a cheek swab and send that. So there's a private companies that do this. You would have to have a really good reason for doing that. Maybe there's a family history, or you're just particularly vigilant, and you'd like to find out if your child has those. So you can certainly do that, but doing that on sort of a population-wide and having the funding to do that, I can't see that being something that any government or jurisdiction would opt to do without sort of a fair reason for that. I can talk to you about, there is an initiative happening in Australia that is government funded called the Mackenzie's Mission, which has been trying to do a blanket sort of a population-wide screening of carrier status. So how many carriers, just people in the room. Okay, this is probably not a good example of just a general population. You're all probably carriers or at some point, but the idea being the general population, how many of us are carrying that, and then would a government sort of prophylactically, preventatively, then look to screen every couple of childbearing age for the carrier status of their, are they carrying these rare conditions? And then would they, if they were at high risk of having a child with a rare condition, would the government then move on to fund IVF, for example, where they can select the embryos, unaffected embryos? Yeah. So that's the ideal, but such a long way off because of the expense, and there's ethics involved, and there's a lot of considerations.
So do the ethical problem, does that come with doing like a whole like screening for everything, or is it just when you do like the targeted screening of rare diseases, is that more feasible?
Yeah, another great question. So in the Australian example, for instance, they took 1,000 rare diseases. So it wasn't just screen for any chink in your DNA. It was, they were specifically targeting 1,000 conditions. But which 1,000? We know there are 7,000 rare diseases and many other genetic conditions that aren't rare. So how do you go about screening those? The BDSRA is in there saying, well, you need to screen all 13 CLN genes. But yeah, it's a tricky one.
Okay.
Ineka, can I speak to that?
Yes,
really
please.
quickly?
Yes.
So I actually was pregnant with both Charlotte and Gwen, and I did the console screen. It's like a fee, like $100, $200. And they did screen for CLN2 and CLN3. So it's when I got the clean bill of goods with both girls, obviously incorrectly, but what they don't tell you is they only tested at the time for, as you said, 100 rare diseases when I was pregnant with Charlotte, and then 200 by the time I was pregnant with Gwen. So I think, Stevie, it's really about advocating for getting all of these rare diseases on a panel so that parents, moms have the option to test in utero. And if they can't get it in time in utero, then just after the baby's born.
Yeah. Okay. Awesome. Well, thank you guys.
Thank you.
I had a question for Christina also, that turn. The ocular gene therapy, is that, so since you guys got like that newly designated regenerative medicine, is that like playing anything like as far as like doing that expanded access, like where you could just go off, wait, instead of doing the FDA thing there, like is there, like when you have a 5-year-old kid, and 5.5, gonna be 6 soon, and has really good vision still, and has got a couple Brianura treatments to the eye, and you're just kind of like, that's like your everything left, is the vision, like last hope. Is there any kind of chance for anything like that with streamlining something, or is it just wait another year or two and just hope that, everything has gone so well with it, obviously, so far. You guys are the furthest along with it. It's amazing. That's my only question.
I expected that question, and I still feel a little tongue-tied in answering it. I'm a pediatric ophthalmologist by background, and my default answer is yes. My purpose for going into medicine and founding this company is to help all of the kids.
Right.
The really awful part is that I can't say yes to everybody, and we're not at a place yet where we can offer expanded access. It doesn't mean that we won't ever, but we're still at a point in the trial where we feel like it wouldn't be appropriate yet.
Okay.
So I'm glad to hear that you're on the IVT Brianura for now.
Yeah.
And
Yeah, we're lucky
hopefully
for
that will buy your child
Yeah.
some time.
That's what we're hoping. Yeah, that's everything that, yeah.
Yeah.
Okay,
Yeah.
and then, I guess one other question too for our friends back there that have a younger guy also with their older one, but can you do that someday, like for the younger guys, like the ocular and the central nervous system? Is that something that could simultaneously be done for them, being that the central nervous system one doesn't help with your vision, right?
Yeah, the central nervous one doesn't help the vision. That's why we're developing separate programs.
Yeah.
And the way that we designed it was to learn about them separately before we put them together, but obviously we wouldn't want parents to be in a position of having to choose one or the other.
Yeah.
So the goal is definitely to do them at the same time if possible, or in the same day, similar to what Inika was describing about the Neurogene program.
That's awesome. Okay, cool. I'm just curious.
Yeah.
All right, thank you.
You're welcome.
My question is for Suzanne Blazier from Lattice. What will your inclusion criteria be for the trial, and how many children do you plan on enrolling? And then thirdly, children who currently receive the Brianura therapy, will they be excluded, or would they have to be off Brianura for a certain amount of time before they can be included? I know these are questions that you probably cannot answer in public, but I'm still asking them. To just give us some idea of timeline-wise when you might be able to proceed with these things.
First of all, thank you for your questions and your interest in the program. Probably my answer is going to be a little bit dissatisfactory. We are working with getting our proposal to the FDA for review and approval. Until then, we are not sure what we plan and propose is going to be, what the actual protocol is going to look like. So I don't want to give hope or give misguided information. And we'll be working as fast as we can to bring it to the clinic. So, sorry not to be able to give you more information.
Thank you.
Hello, I have another question. I know we kind of talked about earlier, a lot of these gene therapies are being delivered intrathecally, and they're just targeting the nervous system, but it sounds like there's still like a pathology in the body, like the rest of the body. So when y'all have looked at the outcomes, like, is that going to be addressed down the line? And if there is a systemic gene therapy given, say you get the intrathecal version, will you have an immune response to a systemic version? Will that child no longer be eligible for gene therapy? And has anyone looked at like addressing both at the same time? Because from what I heard earlier, it sounds like it would be more palliative to just address the nervous system.
Is that?
So, yeah, I think this is going to really depend on the program and the type of gene therapy. Is it an ASO or is it a viral gene therapy, and even the types of viruses that we use. So based on our data and data from many others, the AAV9 actually travels pretty efficiently into the periphery. So we do expect to see targeting of peripheral organs throughout the body, which is a good thing, but can also be a bad thing if the dose is too high, or if you want to dose at a later time point with a drug that's similar, right? So the nervous system might respond to a second treatment if the virus is related, because it has already developed a certain immunity against the first virus. It would depend on how closely related the two therapies are.
Okay.
But on the bright side, we do expect to have some efficacy in the peripheral organs as well, even with the CSF delivery.
That's awesome. I agree with everything that she said. I think that we have to think about safety issues too, because one thing that has been noticed in systemic gene therapy, not necessarily in NCLs, but in other diseases, is that there can be safety issues when you give a gene therapy IV and you get large concentrations in some of the organs, especially the liver. So it seems like very obvious that it's a cookie cutter approach, right? That, okay, so you're giving it to the eye and you're giving it to the brain, and now please give it to the bowel and we'll be good. But it's not quite as simple as that, but certainly the goal is to treat the whole child. But I think it's going to be a more stepwise approach.
So with gene therapy, not thinking about systemically, but just this targeted one, if we do get to a point where it stabilizes disease, are y'all thinking that it's going to be more of like a symptom management, or is the other stuff going to be still like life limiting? Does that make sense?
I think you're going to need to ask a clinician more than the drug developers that question.
Yeah.
I don't know the answer. I don't think anybody knows that answer yet, because these therapies are not far enough along to have long-term data to answer that. I think that symptom management will be important, and I mean, that's what you're doing right now for your children. So time will tell.